So to keep going with Zoom housekeeping, you can ask questions after the talks, and you will be able to do so via the chat box. So we will pick questions from there, but you can also raise your hand and we will let you speak so we can all hear you in here. Yeah, please understand, and that this also applies to the in-person audience, that we won't be able to cover each question. But we do encourage in-person audience to attend the reception afterwards, but also the coffee break. So you will be able to, walk up to speakers and ask the questions there. Yeah, as for the program, which, oh gosh. Okay. So as for the program, we have 2 blocks where we will have presentations, and we will have a coffee break. So this is just a regular break for people on Zoom, but we will serve coffee here in person. And yeah, I think that is about it. And I will happily hand over to Dr. Roberto Flores, who is a postdoc at INSERM 1237 in France, and he will present on mixed pathology and MTL atrophy in Alzheimer's disease.
Cool. So, hi everyone. I'm so happy to be here in person and to be opening this great symposium. Feels good to see you all. And before I start, I would like to thank Laura, David, Anika and Anna for the invitation and for the great organization of this symposium. So yeah, so happy to be here. Thank you a lot. So yeah, I'm going to talk about mixed pathology and MTL atrophy in Alzheimer's disease. So as you all must know, Alzheimer's disease is defined from a neuropathologic point of view by the presence of amyloid plaques, neurofibrillary tangles, and neuronal. And if we are that interested in the medial temporal lobe, is because the MTL is the very first region to be affected by tau pathology in Bragg stages 1 and 2 before tau spreads to limbic regions and ultimately to the neocortex. So, the pattern of propagation for tau pathology has been very precisely described by Bragg and Bragg, and the transentorhinal region is the first region to show tau pathology transentorhinal is roughly located in area 45, which is a subpart of the parietal cortex, right? And then tau spreads to the entorhinal cortex, to CA1, and very quickly to the rest of the MTL. So these early works are very important, but they are also somehow incomplete, since here we only see one slice located in the anterior hippocampus and MTL in general, and we don't have any information about what's happening in the posterior hippocampus or in the parahippocampal cortex. So these days, several groups, including Paul Yushkevich's group at the University of Pennsylvania, take advantage of ex vivo MRI, histology, and AI to build 3D maps that will cover, all the MTL, and that will improve our knowledge of the progression of pathology in the MTL. Another key feature of AD is brain atrophy, as evidenced using MRI. So we can see here by comparing cognitively normal individual and patient with AD that the medial temporal lobe shows very strong atrophy in the disease, and at a more label scale, the atrophy is very strong in the temporal and parietal region. So in this study, Whitwell et al. Followed patients for 3 years, and at baseline, the patients were not demented, and the atrophy was restricted in the medial temporal lobe. And then they were scanned 1 year later, the atrophy had progressed in the medial temporal lobe. And 3 years after baseline, all the patients were demented, and the atrophy had progressed outside of the MTL in the temporal and parietal regions. And very interestingly, this pattern of progression for atrophy is very similar to the pattern of progression for tau pathology. So it really suggests an association between tau pathology and brain atrophy. So indeed, many studies showed these relationships between tau and MTL atrophy using, for example, ex vivo data, where higher Bragg stages were associated with smaller hippocampi, but also using CSF p-tau or tau PET. And it's not only the case for the MTL, but it's also the case at whole brain, and all the voxels of the brain are taken into account, where baseline tau PET uptake is strongly associated with longitudinal atrophy, but it's not the case for amyloid, so that's interesting. So all these studies really suggest that tau pathology is the major driver for atrophy in AD. However, the story is slightly more complex, since additional pathologies are also very commonly observed in patients with AD, including alpha-synuclein and TDP-43. So for example, TDP-43 is observed in up to 70% of individuals with AD, and these AD patients with additional TDP pathology have an increased likelihood of being cognitively impaired at death. So it's very important to take these additional pathology into account, right? Unfortunately, there's no in vivo biomarker for TDP pathology. So the only solution to evaluate the association between TDP pathology and atrophy is to use a combination of neuropathology and ante-mortem MRI. So this is what Josephson et al. Did in several studies. So in this first cross-sectional study, they compared patients, AD patients with and without TDP pathology, and they found that those with additional TDP pathology showed the greater hippocampal atrophy. And they also used longitudinal data and found that patients with additional TDP pathology had an increased rate of hippocampal atrophy than the ones without TDP pathology. So it really shows or suggests that this additional TDP pathology really contributes to MTL atrophy in the context of Alzheimer's disease. However, the contribution of each pathology, tau TDP, to MTL atrophy is still unclear. So we try to answer to this question in this paper published in 2020 in Alzheimer's and Dementia. So we used data in 92 patients with intermediate and high AD pathology, and we had for each of them a neuropathological examination for tau, amyloid, TDP, and alpha-synuclein. So we measured the amount of pathology in 16 brain regions. We could derive an MTL semi-quantitative score from 0 to 3 for each of the 4 pathologies. And we also performed a whole brain staging for each of the 4 pathology, and we also dichotomized 8 patients with and without TDP pathology. So for each of these patients, we also had ante-mortem T1 weighted images where we applied HST1 to estimate the volume of the anterior and posterior hippocampus and the thickness of the entorhinal cortex, BA35, BA36, and the parahippocampal cortex. And we first performed correlations between pathology and MTL structures. And we also compared the patient with and without TDP pathology for each MTL structure. So here are the results regarding the relationship between pathology in the MTL and MTL structures. And we found that the amount of tau was negatively associated with the volume of the posterior hippocampus, where more tau in the MTL is associated with a smaller volume of the posterior hippocampus. And we found negative association between the amount of TDP pathology and the anterior hippocampus, the posterior hippocampus, and thickness of the entorhinal cortex. And only the association between TDP and the anterior hippocampus survived bone Ferroni correction. We also performed surface-based analysis just to explore this association using another technique. And we found that the amount of TDP pathology was associated with reduced volume in the anterior MTL, and tau was more associated with the posterior hippocampus. So we did exactly the same thing using whole brain staging, and we also found that tau stages were negatively associated with volume of the posterior hippocampus, and TDP stages were negatively associated with the anterior hippocampus and the thickness of the entorhinal cortex. So, but also higher alpha-synuclein stages were associated with thinner E36. However, only the association of TDP and the anterior hippocampus survived correction. And the surface-based analysis also showed the same results, with TDP being more associated with the anterior MTL, tau with the posterior MTL, but also with the entorhinal cortex here. Then we compared patient with and without TDP, and we found that those with additional TDP pathology had smaller hippocampus, smaller volume for the entorhinal, anterior hippocampus, and thinner entorhinal cortex. And we also found pretty good AUC for the anterior hippocampus to discriminate the patient with and without TDP pathology, which is very nice since we don't have biomarker now. So using a specific structural measure might be a good target to try to discriminate these populations. So the same kind of analysis were also performed in individuals with suspected non-Alzheimer's disease, so in individuals with SNAP. So these individuals have very strong MTL atrophy, very AD-like MTL atrophy, but they don't have amyloid deposition, so they don't have Alzheimer's, right? And in this paper published last year by Laura, found also an association between TDP43 and tau pathology with MTL atrophy. So to conclude, it really seems that tau is the main driver of MTL atrophy in AD, but there's also a strong contribution of additional pathologies, and especially TDP43 with some specific contributions. So tau seems to be more associated with atrophy in the posterior MTL, while TDP seems to be more associated with atrophy in the anterior MTL. So this in line with other studies, including this one by Yado, published in 2018, where they compared patients with AD and patients in the FTD spectrum, so with probable TDP pathology, and they found that the patient with AD had stronger atrophy in the posterior hippocampus, while those in the FTD spectrum had stronger atrophy in the anterior. So these data also suggest that using structural measures such as the anterior MTL might be relevant to detect additional TDP pathology in the context of Alzheimer's disease, but we really need to confirm that in other data. And there's also probably a contribution of other comorbidities, such as cerebrovascular disease, since we know that, for example, microinfarcts are associated with the volume of the hippocampus. So the story is not complete yet, and we have a lot of different aspects to take into account to really understand the nature of MTL atrophy in AD. So yeah, that's it.
from the in-person audience?
So thank you. I really enjoyed that. In terms of the specificity of I mean, we can go into much more detail in terms of temporal substructure. I think interior posterior is fine, and we think going into a more detailed substructure will give us any more information on how should we look at that in terms of these findings.
To detect specifically TDP pathology or not?
Yeah.
Well, I don't know. We only investigated really the anterior versus the posterior, and it seems to be already good. But now maybe we can try to use different approach to highlight like the very hot spots that could be even more predictive. So this could be done using surface approach, for example, but maybe more precisely than what we've done here. Maybe using ex vivo MRI, for example. So yeah, maybe we might need even more.
And the reason why you selected anterior posterior, was that due to your limited resolution, or was it? Okay. Yeah,
that's the case. So maybe also subfields, for example, might be good. But with, yeah, you're right, with the resolution of these images.
We have one question from the online audience otherwise. The question was, maybe I misunderstood, but tau tangles were not related to ERC atrophy. Is that right?
Yeah, that's right. And that's surprising indeed. So the reason for that is that we define our population based on the intermediate to high AD pathology. So maybe the pathology entorhinal cortex is not like there's not a lot of variability in these regions, since it's already full of NFT in this specific population. So there's other data, including data from Laura and Sadhana Ravikumar from the University of Pennsylvania, using individuals at earlier stages, and they found this association between tau and entorhinal cortex. So it's just because of the selection of our population, I think.
One other question regarding the details. So if I understand it correctly, then your measure of pathology is actually an average across the entire medial temporal lobe, right? Yeah. So it was not like a local relationship of like ERC tau and ERC thickness, or right?
Yeah, exactly. It's MTL, global MTL between entorhinal cortex and CA1, and it's also semi-quantitative, so it's not super accurate too.
Would it theoretically be possible with that data set actually to go into like really even more fine-grained local relationships, or?
Yeah, I think it's something that could be done using digital histology, for example, where you can really have some more specific and more quantitative measure in specific regions. The future. Thanks.
We have otherwise one more question from Zoom. Are there cognitive differences between 80 plus TDP43 versus 80 without TDP43?
Yeah, we did not check that really in this data, but the Joseph's group have investigated this quite a lot, and they found indeed differences in terms of cognition, where those with TDP have more deficits in different aspects of cognition. They are also most of the time older than those without TDP. So yeah, there's a lot of differences between it, not only structural, really.
Okay,
Right.
Thanks all for that.
Thank you.
So next up, we have Dr. J Hodgetts, who is a lecturer affiliated with the Connected Memory Lab at Royal Holloway, University of London. So
thank
the stage is yours.
Do you have a clicker as well?
Yes. Also, just maybe a little note for people asking questions in the in-person audience. We did actually have some issues with the volume. So if you could just speak up a little more, that would be ideal. Yes, yes.
This depends. Is this one here?
Yes, that's.
One in? Okay, good.
So maybe if you find yourself like very far away from a microphone, maybe there is a way to get it to you. Okay, thank you.
Okay, hi everyone. Yeah, so first, thank you to the organizers for inviting me to do this today. I'm actually genuinely excited to be doing an actual in-person talk again. So, and actually to be around the hippocampal family, which is always really fun. So today my talk is the vaguely provocative title, the Subiculum, a hippocampal hub for scene representation. And today I'm basically going to go through, it's going to be a sort of more high level talk where I go through some of my sort of key findings over the last sort of 5 or so years, touching on some of our functional MRI work and also some of our structural MRI work. And basically what I hope to convince you today is that the subiculum has for too long been forgotten. The CA1 has dominated the landscape. So I basically want to rebrand the subiculum for a modern audience. And I hope to demonstrate that the subiculum may actually play a central role in processing visual scenes. So I'm going to start from a sort of broad cognitive perspective, this idea that one of the sort of defining sort of phenomenological aspects of episodic memory is that ability to bring to mind the spatial context in which an event occurred. So if I'm remembering my first cardamom bun in Lund, a large part of that memory will be me reconstructing the rather beautiful cafe in which I consumed it. And often these spatial representations are not often single snapshots like photographs, but something much more flexible and integrated where we can often imagine different viewpoints, we can plan paths or routes through that space, and imagine different relationship between objects and people. And in recent decades, this idea has been extended to the extent that several theoretical models have emerged, which have highlighted scenes as being central to several interrelated cognitive functions. So not just episodic memory, but also navigation, visual perception, and also imagination and future planning. In line with this, there seems to be overlap in the actual neural substrates of these different functions. So there's been work done showing that patients with damage to the hippocampus show scene-related deficits across a range of different tasks. So work conducted by Andy and colleagues in the early 2000s, for example, showed that patients with acquired hippocampal damage or with Alzheimer's disease are impaired at discriminating between scenes presented from different viewpoints. Other work, for example, from Maureen Irish in 2015 showed that Alzheimer's patients also have problems imagining or constructing scenes. And then also work from Karen Taylor and colleagues in 2007 showing that patients with hippocampal damage are impaired when trying to recall scenes, particularly from different perspectives. And this has been shown across a range of other different functions as well, so including working memory and short-term memory and so on. So a lot of the work that I did initially was really trying to see whether we can find strong evidence for this using functional MRI in healthy participants. So what do we do in the first study? Well, we did a kind of large-scale fMRI analysis. So we had around 50 subjects who all completed this very basic end-back paradigm. So they just viewed blocks of scenes, which are shown on the slide, but also objects and scrambled objects. And participants just simply had to indicate when an item repeated. And these stimuli are only presented for sort of 400 milliseconds. So the demand on long-term memory is very limited. But what's particularly interesting about this is we focus not only on the hippocampus, but also on this more extended scene processing network. So what we wanted to do was compare the functional properties of the hippocampus in this task to also the posterior parahippocampal gyrus, or PPA, the retrosplenial cortex, and also the transverse occipital sulcus, or the OPA. So first, we did a standard group level analysis. So when comparing scenes versus other classes of stimuli, we found that this extended network is robustly activated, including PHG, RSC, and TOS, but also that we get strong activity in the anterior medial part of the hippocampus. Evidencing this further, what we wanted to do is also see, well, actually, when we look at individual participants, how many of those are actually activating the hippocampus robustly at the individual level? And what we find is that the hippocampus, in 90% of participants, 90% of participants activated robustly their hippocampus when looking at scenes versus other types of stimuli, to the extent that it was actually greater than seen in other so-called core regions, such as the retrosplenial cortex. But what was really interesting is actually when we started to map individual level activity. So what we did is we took individual scene clusters and we just plotted the peaks of these within the hippocampus. And what we found is two things. Firstly, that these peaks are slightly variable across participants, which is interesting, but also that they tend to converge on this very specific anteromedial part of the hippocampus. And this is interesting because it converges with a whole body of work that was also reviewed by Simon and McGuire in 2015, which shows that you get this similar anteromedial hotspot appearing in several tasks that involve the perception and imagination of scenes, but also in tasks involving navigation, autobiographical memory, and so on. So this is an interesting study in two ways. First, we show that the hippocampus shares many functional properties with this extended scene network, but also that its role in scene processing may not be a property of the hippocampus as a whole, but rather of a particular subdivision located in the anteromedial part. So how can we dig into this even further? So as we all noted, it's the hippocampal subfields group. So we know that the hippocampus is not just like one lump of gray matter. It's more like a patchwork of different subregions. So we can subdivide the hippocampus based, for example, on cytoarchitecture. So we obviously know from histology that the hippocampus contains all the different subfields. So we have CA1, 2, and 3, dentate gyrus, and my old friend subiculum as well. We also have functional organization that varies along the long axis, along the anterior to posterior axis, which I'm sure will be discussed today, and Robin touched upon this as well. So we get variation, we get these connectivity gradients along the long axis, and then also work in fMRI and also in electrophysiology, showing that the cellular dynamics of place cells, for example, vary along the long axis. Complicating matters further, we also have variation along the transverse, so the medial to lateral axis, and also along the kind of proximal distal components of different subfields. So proximal and distal CA1 and subiculum, for example, project to slightly different regions and also form distinct circuits within themselves. So we have a pretty good idea of how these things work in non-human species, using sort of axonal tracing and so on. But it can be quite challenging to look at this stuff in humans. So one of the ways we can get at this is we can use like high resolution fMRI to get us closer to kind of uncovering this kind of more fine-grained functional organization. So this is what we did. We used 7 Tesla MRI to examine the role of different hippocampal subfields and also these long axis subdivisions in perceptual discrimination for scenes versus other categories of stimuli. And in this figure, it's just showing the kind of gains you have from 7T. So the top one is actually from the 2016 paper, where the hippocampus is just a gray mush. And then as we move to panel B, which is what we use in this task, we can see that we can already start to see some anatomical structure within the hippocampus. And then more recently, and I know we'd work done in DZ&E, there's movement towards using like 0.8 isotropic voxels in functional MRI. So what we did, we had participants complete this odd one out perceptual discrimination task, which had been used plenty of times in patient and lesion studies, where participants have to find the odd one out when looking at these arrays of scenes, spaces, objects, and also these this baseline condition of size. And in these same participants, we also collected this ultra high resolution structural data, which allowed us to manually segment the different subfields. I found ashes after I finished doing this. So that was a helpful moment. So yeah, so I manually segmented that, which was fun. And then when we extracted the parameter estimates for the different for these different stimulus types, what we find is that the subiculum is the only region that shows modulation by category type. So the subiculum shows significantly stronger activity for scenes, versus faces and objects. And what was interesting is actually when we broke this down further is that this was unique to the anteromedial component of the subiculum. So again, we're just refining what we showed in that 2016 paper showing that the hippocampus's role in scene processing may actually be something unique to the subiculum and the anteromedial subiculum. Right, so I'm going to sort of zoom out a little bit now. So as I alluded to earlier when talking about our earlier paper, the hippocampus is not an isolated thing. It actually forms multiple circuits, and it's part of an extended system. And what's particularly interesting about the subiculum is that actually most of the hippocampal outputs actually emerge from the subiculum. So maybe we can actually rebrand this as the extended subicular system. And what I show here is that actually hippocampal outputs to several structures that have been implicated in scene and event cognition, such as the mammillary bodies, anterior thalamic nuclei, and the retrosplenial cortex, emerge almost entirely from the subiculum, so that's shown in green here. Whereas outputs to medial prefrontal cortex and parahippocampal cortex still emerge predominantly from the subiculum, but they share outputs with the CA1. So how can we actually start to look at these different circuits? One way of doing this is not actually to look at the properties of the individual brain regions, but actually to zoom in on the white matter itself. So what we can do is use diffusion MRI to study the properties of this. And I've isolated the fornix in particular because this is mainly comprised of subicular projections and terminates in mammillary bodies, anterior thalamic nuclei, and the medial septum. And this is just, top right is just showing a general idea of the approach. So we can go into, can collect diffusion MRI data and we can reconstruct fornix in vivo. So you get these kind of really kind of cool images of the fornix. And then we can do some modeling and derive metrics that reflect the kind of microstructural properties of this tract in each participant, which hopefully have some relationship with cognitive performance. Okay, so in the first study examining this, what we did is we took the same test that we had in the Journal of Neuroscience paper. So this is this odd one out discrimination paradigm that had also been shown to be impaired in hippocampal patients. And what we wanted to see is whether fornix microstructure was more strongly related to performance in the scene task versus the face task. And then what we also had is we compared this with the this long ventral pathway here called the inferior longitudinal fasciculus, which provides inputs into the perirhinal cortex and anterior temporal lobe from early visual areas. And indeed, this is what we found. So we saw that the microstructural properties of the fornix was strongly correlated with scene discrimination accuracy, whereas individual differences in the properties of the inferior longitudinal fasciculus was more strongly related to face performance, which is shown in the bottom left of this figure. So we've shown that in perception, but what about memory? So in this task, we collected diffusion MRI data in a bunch of participants who completed a kind of autobiographical memory task. So here they are given keywords and they have to generate an everyday memory associated with that. And what we can do is we can use Brian Levine's autobiographical interview coding method to extract the kind of spatial episodic information and the semantic information from these stories. So the question we posed was, well, is the fornix also related to kind of the degree at which people produce spatial episodic detail in just their everyday memories? And basically that's what we found as well. So this is just showing the regression coefficients for both fractional anisotropy and mean diffusivity, so the two diffusion MRI measures. And what we found is that the fornix was strongly related to the amount of spatial episodic detail that people produce, but not semantic information. And then again, we saw the inverse for the inferior longitudinal fasciculus. Also in a study, more recently a study by Ang Williams and stuff, we built on this further and looked at different fornix subdivisions. So if you're interested in that, I'm happy to talk about it. So how are we doing? You might have held up. Okay, cool. So I'm just going to sort of quickly summarize. So what I hope to have demonstrated is that we can, by doing sort of more fine-grained mapping of functional activity, we can look in more detail at functional organization in the hippocampus. And this work demonstrated that the anteromedial subiculum may have a very precise role in complex scene processing. Further, I showed that the fornix, which is mainly comprised of these subicular output fibers, supports scene perception, but also contextual aspects of episodic remembering. And generally what I feel is that the subiculum basically has a special position in the hippocampus. It is at the interface between the hippocampus and the rest of the brain, and via its extremely strong dense connectivity with several regions implicated in spatial processing and memory, that it has a sort of unique functional significance and deserves more interest in the community. I'll skip over this because there's a lot of detail. But there are lots of different questions to be answered, and that's kind of what we're starting to do now. So I've got a new postdoc, Sam Berry, whose background is more in sort of functional anatomical connectivity, and he's just started a project looking at trying to characterize subicular connectivity using high resolution diffusion MRI data, and also examining whether using kind of graph theory to see whether the subiculum does act as a hub within this extended network. We also have Lucy Reed, who's developed a new task to examine the precise role of the subiculum in scene perception, with a particular focus on a potential role in integrating different scene views into these kind of higher level representations of space. And then we also have two PhD students starting. One, Kavishni Appasamy, is going to be looking at hippocampal cerebellar interactions. And then finally, Eleanor Alderman's going to be doing some cool work looking at scene vision and scene exploration and how that relates to episodic memory. So it's exciting times. So yes, thank you to all these people. They've been amazing. So thank you for listening.
Thank you so much. Any questions from the audience here in person?
And I hope I remember this correctly. In the start of your slides, you said that you, like really at the start, you had a talk, like a finding about scene perception, that 90% of people have activated hippocampus versus scene test. So what about the 10% that don't have that? What's up with those?
Yeah, that's a good point. I mean, I guess it's quite possible there'd be sub-threshold activation. Obviously, it's, you know, with signal to noise, it's actually almost surprising that we saw as many as we did, knowing that, you know, SNR is so low in the medial temporal lobe. I mean, interestingly, in the 7T study, we never published that in the study, but we actually did the same analysis again and 100% activate. So it could actually be a sensitivity issue in the methods that we used. I mean, I don't think it's 100% like PPA. So you're going to expect some participants that don't activate, maybe, you know, they're just thinking about something else or asleep or something. But yeah, so it could be a signal SNR kind of thing. But it's way higher than maybe we anticipated, given those limitations.
Great. Following up on Laura's question, what was the contrast for that? Like, because you had kind of mentioned that there was more activation of hippocampus scenes compared to objects, but what was like the baseline or?
Yeah, so that was in the kind of consistency analysis that was scenes greater than objects, but we showed it for scenes greater than objects and also greater than scrambled objects as well. So we demonstrated across all kind of scene relevant contrasts. I think ideally you probably want more stimulus classes in there so you could kind of get a more nuanced picture of what's happening. But yeah, so they were the contrasts.
And so the scenes were like, sorry, just looking over the scene information. So in terms of those scenes, those kind of seemed like more like bare bones scenes, like there weren't really a lot of like objects and like context in there. And do you think that if you followed that up, in which, like, you know, you did scramble scenes and there are objects within the scene, so it's hard to think of this object versus scene processing?
Yeah, that's a really good point, because I mean, in the 2016 paper, yeah, we used these kind of real world stimuli, and then obviously for the 7T study, we used these kind of virtual reality spaces, which are very kind of bounded. Yeah, I kind of agree. I'm kind of interested in actually what are the underlying properties of space itself that's driving this, because just to say it seems like, what does that mean? Is it a specific property such as boundary or bounded spaces? That's particularly interesting to me in the context of the subiculum, because we know it codes for allocentric boundaries and so on. So it'd be interesting to look at how that varies across different scene types. So that
you get an association between like PPA or something and like subiculum where this integration is happening more.
Yeah, maybe that CA1's doing stuff that we don't realize, right? If we probe it correctly. Yeah. So yeah, it's a good question. Hi.
Hi. I have a question for your high-res 7T study. Did you also look at entorhinal cortex?
No, but that was, we really wanted to, but basically just signal was dreadful. So yeah, we just had really bad SMR and ERC. So yeah, the kind of sequences we're trying to develop now, we're trying to just kind of boost that so we can actually look at ERC in more detail. I think, can't remember, maybe it was one of David's studies or something where they saw similar effects across medial entorhinal and medial subiculum. So they may share similar properties, but maybe that's a spatial resolution thing as well.
Okay, we have a bunch of questions now coming in. I'm just coming closer so they can also hear the question. Does your enteromedial subiculum include the pre-parasubiculum? That seems to be the case from the two images you showed with your delineation of subiculum.
Yeah, so I kind of knew this question would come up. So so one of the things we haven't done in these studies is discriminate between pre-parasubiculum and subiculum proper. It's partly because actually I don't know who asked the question.
No.
Okay, well I don't know if the partly because there's not an extremely well validated protocol for distinguishing between those two things anyway on structural MRI. That's something we kind of want to do now with this new project grant. But I think it'd be really interesting to compare medial subiculum, pre-subiculum. It may that it is actually more property of pre and parasubiculum versus medial subiculum per se.
Yeah.
Particularly as that's has much stronger connectivity with say retrospinal cortex, which maybe has a similar kind of functional role.
So he's happy.
Okay, great.
How does your delineation of the anterior medial subiculum relate to the proximal distal plane of the subiculum?
Yeah, I guess that's a kind of similar question.
Yeah.
It's hard to, again, it'd be hard to know
exactly
what. Yeah, I mean, it'd be interesting to do that in a sort of you could extract those potentially using like connectivity as a parcellation method potentially. That'd be quite interesting. And that's something we're trying to look to do now. I sort of showed it very briefly. It does seem that you can distinguish two halves of the subiculum in the anterior aspect based on its structural connectivity. So that might be one way of getting at that kind of more distal thing. So rather than using landmarks, actually use connectivity data.
Yeah. Okay.
It's a provider.
Good question. To what extent do you think the subiculum is involved in contextual integration more broadly and scene perception as one situation where contextual integration is at its maximum?
Okay.
Here.
Yeah, I think I probably agree with that to some extent. I think it's probably, although in the 2016 paper, we used these singles viewpoints, we obviously saw much stronger effects in the 7T study, where we did show scenes from different viewpoints. So I do feel that the integration is a key aspect of it. That's something we want to do, is like looking more at how exposure to scenes from different viewpoints where you have to actually integrate information effectively in the task, whether that is more of a driver of subicular activity than just I'm perceiving a scene. But it also might be, like, as the question was asked earlier, about the properties of space individually, so things like boundaries and things like that.
Yeah, I can just read out. You mentioned that there is an anterior-posterior gradient of place cell responses in the hippocampus. Do you think this may be related to scene processing selectiveness?
It's possible. I mean, I guess it's vaguely complicated by the fact that that gradient, I'm not sure to what extent that gradient holds across different subfields as well. I know that those studies were mainly done in CA1, but then subicular place fields also differ to CA1 quite dramatically as well. So I think CA1, subicular place fields in general are much larger. They tend to spread to cover whole spaces rather than being more fine-grained, if I'm remembering the work from like Patricia Sharp and so on. But that is kind of consistent with some of this stuff, again, about it's more about creating these more general contextual representations than purely a single scene view. But yeah, I'm completely speculating how it links to play cells, but
yeah. Okay. Then we have one more question from Menno. You mentioned that you looked at subdivisions of the fornix. Am I correct in assuming that the stronger association between fornix and scenes is stronger in the post-commercial fornix? That indeed mainly carries subicular output axons, whereas the pre-commercial components has many CA1 and subicular axons.
yeah, so interestingly, we didn't look at that in relation to the scene perceptual performance, but we did do that in relation to the autobiographical memory task. And actually what we found it was stronger for the actual pre-commercial component. So the part that is more strongly connected to one that links to the medial prefrontal cortex. But that's partly because it's actually very tricky to reconstruct the post-commercial component as well. So they're the bits obviously like dive down more to the and so on. Or it at least combines those thalamic projections within the memory bodies, and that kind of gets more complicated. So yeah, we want to kind of, there's also kind of medial lateral fornix as well that also differ in terms of anterior versus posterior hippocampus, and that could be kind of interesting as well. But again, we're kind of working on new protocols for doing that. Yeah, interesting.
Yeah, okay. There were all the online questions. I don't know if there are more here.
All right. Well, thanks a lot again. So next up, we have Dr. Kristin Nordin, who is a postdoc at the Aging Research Center at Karolinska Institute. And she will talk about topographic organization of cortical connectivity along multiple hippocampal axes in aging.
Yeah. All right. So how?
Oh, yeah, sorry.
No, I think it's fine. Thanks. All right. Hi, everyone. I also want to start by saying thank you to all of the organizers for putting together this symposium and for having me here as a speaker. My plan is to present some results from an ongoing project because I thought this is the best opportunity for showing some under or in progress hippocampal data. This is a project in which we're looking at the organization of functional connectivity with the cortex within the hippocampus across the adult lifespan. And I want to start off in this gradient mapping or from a gradient mapping perspective. So gradient mapping methods to characterize regions' functional organization are becoming increasingly more common. And there are several studies in younger adults showing that connectivity is organized along multiple overlapping axes within the hippocampus. And I would say that the most common finding across these studies is that the dominant mode of connectivity is organized along the longitudinal hippocampal axis. And there are more examples than these. And I think it's important also to kind of mention that there are a lot of different modalities that seem to map onto such functional gradients. So the organization of structural covariance, molecular organization, and also the way in which cognitive domains fall onto this axis. But we are interested also in aging. And even though we know that there is variation in large scale connectivity along the anterior posterior axis, also in older age, this type of gradient mapping approach is less common in aging, and especially in terms of assessing individual differences in gradient properties and how they relate to, for instance, memory or age. And what we're looking in our studies also dopamine. So we're using the dynamic study. It's a data set we have at Aging Research Center and also at UFBI in Umeå. It consists of 180 adults, evenly distributed across 6 decades of the adult lifespan. And we have a lot of data from these individuals, and it's resting data from our data that we're using in this study. And also we have sharing PET data for all of these individuals, which means that we can assess the availability of the dopamine D1 receptor in the brain. And the gradient mapping method we're using, it's connectopic mapping, which operates on the similarity across voxel-wise fingerprints of cortical connectivity within the hippocampus, and identifies these connectopic maps, or in other words, gradients. And what we like about this method is that in a final step, there's an implementation of spatial statistics onto these gradients that try to model the spatial characteristics of each gradient for each participant, to describe each gradient in a smaller set of parameters. And then we can use that set of parameters for each individual, for each gradient, to look into individual differences in gradient characteristics. That is what we're doing. That's what we've done. And I want to talk to you about 3 main gradients that we find. So consistent with previous studies, the main gradient, or the principal mode of organization that we identify, is this longitudinal anterior to posterior gradient, in which we see.
Okay, everyone. Sorry for that unexpected interruption. We are back. People on Zoom can hopefully see us. Otherwise, please write that in the chat box. However, it should look good. So I would suggest we just continue where we stopped, if that's okay by you. So yeah.
Okay. All right, so I was just about to talk about these 3 gradients that we identified in our adult lifespan sample. The first gradient showing this gradual anterior to posterior variation in connectivity patterns across the hippocampus, whereas second gradient was also a longitudinal axis gradient, but instead showing variation in connectivity from middle parts of the hippocampus spreading towards the anterior and posterior ends. And then third order gradient, in contrast, showing a medial lateral pattern and not very much variation along the longitudinal axis. And I'm going to say a few words on the effect of age on these gradients, but then I'm going to mainly focus on the first principal anterior-posterior gradient. All right, so using these gradient parameters that we get from the trend surface modeling, we assessed age, continuous age, as a predictor of these gradient parameters. And we can see that gradient parameters of all 3 gradients showed age related variation. And the strongest effects were found for this first hippocampal gradient. And in addition to age, so what we did then to kind of visualize this was to divide this gradient into bins of about 2 millimeters and plot the average connectivity value for these bins as a function of their distance from the most anterior voxel in the hippocampus. So what we first of all can see is that this type of variation, anterior to posterior, is quite graded and smooth along the axis. And we see it in all of our participants, basically, or independent of age. So it's evident in the younger age group, middle-aged, and also in our older segment. However, we know there's an age effect, and one way in which we interpret this age effect is that the dark purple curve that you see is the older age group. They are from 60 to 80 years old. They show like a flatter curve or less distinct change in connectivity along this axis. And then, as I was saying, in addition to age, we were also interested in dopamine, because we know that dopamine is important for processes in the hippocampus. It decreases as a function of age. A lot of dopamine markers do, which has an effect on cognition. And the dopamine modulation is also important for the distinctiveness of neural representations. So we wanted to know if dopamine D1 receptor availability had some sort of effect on the properties of this first gradient. So when we looked at all of our participants at once, controlling for age and motion parameters and so on, we could see a significant effect of dopamine on the spatial characteristics of this gradient. When we split our sample into our 3 smaller age groups, we see that this effect is only significant in the older age group. On the other hand, in terms of episodic memory, using gradient parameters as predictors of memory performance, we see that it looks a bit blurry, but all right, we see that gradient parameters seem to predict episodic memory performance in younger adults, and this is true for the left hemisphere gradient. And just in terms of younger adults, this is consistent with what they've shown in a previous study in younger adults using connectopic mapping on the hippocampus or in the hippocampus. But then we wanted to know, okay, so this gradient is also evident in middle-aged and older adults. It's age sensitive and it's related to dopamine in older adults. So is there a functional relevance of this gradient in aging? So we used latent class analysis to see if we can identify groups of older adults that differ on the gradient properties of this first principal gradient. So using left hemisphere gradient parameters in an LCA, we could identify 2 groups of older individuals, one which we call youth-like, or older adults with a youth-like gradient or more youth-like gradient, and older adults with a more aged gradient. So these 2 groups obviously differ significantly from each other in terms of gradient parameters. Both of them differ from younger adults in terms of gradient 1, that is aged group, to a greater degree than the youth-like group. And interestingly, even though no data from gradient 2 or gradient 3 went into identifying these groups, we see this difference in relation to the gradient in younger adults in this aged group, also for gradient 2 and 3. Okay, so to the left, you see gradients computed at group level for these two older groups. And these plots show also the differences between these two groups in the change in connectivity patterns along this axis, and show quite nicely that this youth-like group follows the youth or the curve in the younger adults to a greater extent. And importantly, we wanted to know if this have any bearing on episodic memory. And while they only differed at trend level for our composite episodic memory score, which consists of word recall, number word associative recall, and object location recall, they did significantly from the word recall subtest specifically. They did not show any difference in gray matter volume, nor in average level of D1 receptor availability. And there was a significant effect of D1 on gradient properties in both of these older groups. And they did, however, also differ on white matter lesion volumes, suggesting maybe some vascular dimension to this grouping. But yes, so one remaining question that we're pursuing right now is to dig deeper into a potential role of dopamine in gradient maintenance in aging. So if these results suggest that having a youth-like gradient, a maintained anterior-posterior functional gradient in older age is beneficial to memory, does dopamine play a part in that? And for that, we want to move away a little bit from these average levels of D1 receptor availability and look into options of characterizing the potential overlap in the spatial distribution of D1 receptors, how that would map on to the topographic organization of our gradient. Yes, so to the summary and conclusion. So what we find is that the principal gradient of cortical connectivity is organized along this anterior posterior axis across the adult lifespan, but that the spatial characteristics of this gradient show significant age-related variation with less distinct transitions in connectivity along this axis. And also that a greater youth-like gradient seems to be beneficial to memory performance in older age. And if I have like a minute left, I can just show you that we're also pursuing gradient 2 and 3 more, and wondering how they might fit into what's been previously reported. I'm not going to touch anything but one specific button. So surprisingly, our gradient 2 was show the same pattern that we've reported before in an older sample, but using a parcel-based approach. So we found components of connectivity that distinguished between the anterior and posterior regions, and a second significant component separated the middle hippocampus from anterior and posterior hippocampal regions. And a similar gradient has been reported as a third order gradient in younger adults. And then our third gradient is this medial lateral gradient, we think, which often emerges already at like second order positions. And then I also just want to acknowledge that there's a group of us working on this project, both at Aging Research Center and also at UFBI in Umeå. So thank you very much.
Also, again, sorry for the technical difficulties. I'm glad that it went through like this now. So any questions from the in-person audience to start with?
Yeah, thanks for your talk. If I recall correctly, you showed that only the gradient of the left hippocampus in the young adults was related to memory performance. So do you have any hypothesis on why not the right?
I don't know the clear hypothesis. So the memory performance score was a mixture of different, not modalities, but different types of content. So it's not a clear verbal task, for instance, and it didn't have a very strong spatial component either. So I think what they found before was that if they compared faces to scenes or no, I don't know honestly what that would be.
Look at overall hippocampal gray matter differences and you didn't find any differences. When you split it into left and right, do you see volumetric differences?
No. So I should have mentioned that this was left side, but it's the same left and right. It's the same bilaterally. And we've also compared these two groups on anterior and posterior. They are quite similar.
The next up, thanks, sorry, thank you so much for the talk. So next up we have Niklas Vockert, who is a PhD student at the German Center for Neurodegenerative Diseases in Magdeburg, Germany. He will talk, oh, I'm sorry, thought I could do this by heart. He will talk on hippocampal vascularization patterns and their association with brain structure and cognition in old age.
All already. Yeah, thanks a lot, Hannah, for the kind introduction and also for organizing all of that. And for giving me the opportunity to speak here and present my and our lab's data on exactly this topic. Yeah, I'm basically representing Anika here today, who's my supervisor, and she says hi to every one of you. Alrighty, so let's get into hippocampal vascularization patterns first. So there have been two seminal post-mortem studies in the past, almost 30 years ago, that have shown that there are mainly two arteries that are involved in hippocampal blood supply. And the first one is the PCA, it's shown in blue, that's like this one here. Oh, it's hard to do that. And this has been found to supply the hippocampus with blood in all of the examined hemispheres. On the other side, there is the anterior choroidal artery, or ACHA, that is always present, but only sometimes additionally contributes to hippocampal blood supply. And if it does so, it primarily supplies the hippocampal head. So you can imagine that a given individual has either 0, 1, or 2 hemispheres where the ACHA additionally contributes. By now, fortunately, we can do that in vivo and don't have to look at deceased people. For that, we use 7 Tesla MRI with a so-called time-of-flight magnetic resonance angiography with a resolution of 0.28 millimeters. And there was a study at our institute, even before I joined, of Marco Spalazzi, where he showed, where he basically established this and found that there were similar proportions of hemispheres with the ACHA contribution compared to the post-mortem studies before. So kind of showing that this might be a valid approach to actually assess them in vivo. And as I said, there are different possibilities. So on an individual level, you can imagine that a person has 2 hemispheres where only the ACHA contributes, and we would call that a basic supply. And I want to contrast that with an augmented supply, where there's at least one hemisphere with an additional contribution of the ACHA. And to introduce our sample, as the title already suggests, we're looking at older adults, 47 of them that were about 71 years old. And the important part is that 20 of them have vascular pathology, namely cerebral small vessel disease. And this is a disease that comprises several diseases that all affect small vessels in the brain. And by small here, I mean less than 1 millimeter in diameter. The clinical phenotype of that is that those people have an increased amount of hippocampal microinfarcts and atrophy, more neuronal loss, and a decrease in regional cerebral blood volume. And of course, for these participants, we have our 7T TOF that we use then to assess the hippocampal vascularization patterns, which was successful in 43 subjects. So a previous colleague of mine, Valentina Perosa, also shown on the slide, she examined the association of the HVP with cognition and found that on various cognitive tests, people with an augmented supply, here shown with a 1, they had better cognitive scores. Please note that for the ADAS-Cog, a lower score means less errors, so better cognition. And so we have 2 more cognitive tests and 1 that is a bit more memory-based, the California Global Learning Test. Then this is probably what you're most interested in here, the associations with local structure in the MTL. So for this, we did the processing with T1 ashes because we didn't have a dedicated high-resolution T2 sequence, as Robin already introduced. So we got the entorhinal cortex, the anterior hippocampus, posterior hippocampus, perirhinal cortex, and parahippocampal cortex. And we did a maneuver then to see if an augmented supply would be related to bigger anterior MTL volumes or posterior. And we found that an augmented supply only was associated with greater anterior medial temporal lobe volumes, but not posterior MTL volumes, which matches the specificity of the blood supply of the ACHA to the hippocampal head. And we further found that in post-hoc analyses that this is mostly or partly driven by the anterior hippocampus and actually mostly apparently by the entorhinal cortex. In terms of CSVD, there was no difference between CSVD patients and controls. Strikingly, this seeming effect extended beyond the local structures towards the global brain structure. So we also found that an augmented supply was associated with higher total gray matter volumes in the whole brain, and also CSVD patients had smaller total gray matter volumes than controls. So there were main effects both of CSVD status and the hippocampal vascularization pattern. So we did a mediation analysis to tie all of that together, and the results showed us or are in line with our hypothesis that an augmented supply leads to greater total gray matter volumes that then in turn lead to better cognition. So the total gray matter volumes are the mediator here. Additionally, we wanted to go a little bit beyond these mere binary vascularization patterns. So we teamed up with an engineer, Henrik Mattan, and looked at something called vessel distance mapping. And our nuclear physician, Berta Garcia, she currently submitted a paper on this. And for this, she took the 7 Tesla TOF image and manually segmented arteries, which is a whole lot of work. And we're going to focus here on the PCA and ACHA because only those are the ones that are important for us. And I'm just going to go over these 3 metrics here very quickly in the interest of time. But there's global VDM, which basically looks at single voxels of the hippocampus and how far they are from their closest vessel irrespective of which vessel it is. And then to obtain one measure for an individual, we just take the mean across the whole hippocampus. VSVDM, or vessel-specific VDM, looks at this like at the distance of the hippocampal voxels to the specific vessel, so to the PCA and to the ACHA, and then combines that in something like an average. And at last there is the center of mass distance, which is most similar to the vessel pattern, while the other two metrics are more representing vessel density, you could say. And there you only look at the main trunk of the two arteries, so the PCA and ACHA, and consider the center of mass as well as the center of mass of the hippocampus, and calculate their distance, and again, combine that in kind of an average. Berta then looked again at the associations of these metrics with cognition, and it's only exemplary shown here for global VDM, but it's really the same for all three metrics. And she found an interaction effect between those metrics and CSVD status. And what that means, you can nicely see here on that graph, where the blue points are the controls, and you can see that in the controls, the VDM metrics don't really seem to influence how good cognition in those patients, or not patients, in these controls actually is. In contrast, if you look at the red points, the CSVD patients, you see a clear relationship between VDM and the cognition. So higher values of VDM, meaning greater distance to the closest vessel, means greater ADAS-Cog scores, so worse cognition. And this actually fits to also what Valentina found earlier, where with the patterns again, she found that if you look closer, actually the people that like the controls, they don't differ based on the hippocampal vascularization pattern in terms of cognition, but then the people that have CSVD, so vascular pathology, there the augmented supply really seems to bring a benefit for cognition. One additional thing we wanted to look at is which of those metrics is actually the most important one for cognition. And here we just consider the variance in ADAS-Cog explained by a simple model with an interaction term between the metrics and and CSVD status, as well as the main effects and some covariates. And we found that actually the center of mass distance, the ACHA radius, and the global VDM seem to be the most important metrics here. Yeah, to wrap that up, the HVP seems to exert both global and local structural effects. As I showed you, it is associated with higher gray matter volumes in the anterior MTL, but not posterior MTL, which nicely fits the specificity of the ACHA supply. Interestingly, the entorhinal cortex seemed to be the region that benefits the most, which is interesting because it's, as we also already saw, is one of the earliest regions where tau accumulates. Then more on the global level, there seem to be more far-reaching implications. So the total gray matter is also increased in augmented supply individuals that then produces better cognition. And there are like two hypotheses here, that either the vascularization in other brain areas co-varies with the hippocampal blood supply, or simply that the increased potential for hippocampal plasticity might then have an effect on cortical and subcortical networks. Just overall, the hippocampal vascularization pattern could present a factor, or just general hippocampal vascularization could present a factor for brain reserve or brain maintenance. And with this study that is just cross-sectional, we unfortunately cannot disentangle those two. And it also seems to be implicated in resilience. Yeah, this is a very simple framework and to the most extent even just binary, but I want to stress again that this is still quite valuable and could help to identify individuals that are early on that are at risk for stronger cognitive decline or dementia. Yeah, what we want to do in the future and what we're actually doing right now is to look at the same thing in young adults, which would then allow us, for example, to look at the distinction between brain maintenance and brain reserve. And there we want to specifically also relate the HVP and vessel distance markers to blood flow measurements that are more quantitative, cardiovascular activity that we can assess with resting state fMRI, and hippocampal and MTL activation during a memory task in the fMRI. With that, I want to thank everybody who was involved in the project, and especially Valentina, Berta, Hendrik, and Anna, and also you for listening. I'm open to questions now, or just meet me in the coffee break.
Thank you so much for the fantastic talk, Niklas. As before, we will start with questions from the in-person audience. His hands, yeah.
Thank you so much for your talk. I was wondering if you could comment a bit more on the augmented group. So was there, what was the distribution like just within that group? Was it the case that most people who had an augmented vascularization had it to both hemispheres, or was it quite distributed between the groups? And was there any dose-dependent advantage of having it for both hemispheres as opposed to just one?
Yeah, that's an interesting question. So it's roughly, you can say, a quarter each of the 4 possibilities that I've shown. So basically then 25% of what I would call a twice augmented supply. And then around 50% once augmented and 25% basic supply. And actually, just looking at the data, it doesn't really seem like there's a huge additional benefit to having the second hemisphere also with what we call then dual supply, so with the additional ACHA contribution. It seems to be relatively equal between the once and twice augmented group. Yeah.
So I'm interested in the longitudinal stuff that you're about to do. So I would just like to hear, can you speculate a little bit about, would you expect the person, for example, having started with dual supply, changing to single supply, or how stable would this vascularization pattern be across the lifespan? And when would be the sweet spot for a change?
That's also something we've been wondering about. I personally think that this is something that would be rather robust in general. I assume there's totally some angiogenesis on the smaller level, but that the vessel pattern really changes. I think that is something that is, if anything, rare, I'd assume, but we don't have any data on that.
Have you looked at, or are you planning to look at whether having the augmented supply is related to protection against developing tau pathology in the entorhinal cortex?
We just looked at this in relation to vascular pathology, and there was no benefit in terms of resistance, actually. But in Alzheimer's disease, we don't know that yet. That would also be something very interesting to look at. Yeah.
I was curious, do you know if there's like another brain region that has like this augmented supply option or something to check that?
Yeah, I don't know too exact, but I know that other people in our institute are, for example, looking at the basal ganglia and the motor cortex, and there are also differences. I don't know if it's exactly like augmented and basic, but there's also some variability in the blood supply that we're looking at.
All right. Yeah, thanks a lot again. We will now proceed to to have a coffee break, and we will have to stick to our original schedule. So we will continue at 3:05. It's now 2:45, so we have 20 minutes. To the online audience, I would really appreciate if you would stay logged in so we don't have to readmit you to the meeting. That would be great. Grab a drink wherever you are right now, and we'll see you again at 3:05. Thanks.
the hippocampal organization is that it is divided into subfields based on its cellular subdivision and composition. So, and this is mainly done in 2D, but if we reconstruct it back into 3D, we still see a very prominent medial lateral differentiation, one subfield following the other subfield. However, we can also describe hippocampal organization in the anterior-posterior dimension, which was done based on macroanatomy, function in rodents and humans. So, so far what we know is that we have actually a lot of knowledge how the hippocampus is organized based on its site architecture. But we were lacking at this time a differentiation pattern based on large-scale networks. And this is what I did in my first study. So I had 3 different connectivity measures. The first one was meta-analytical connectivity modeling, which describes the co-activation pattern of the hippocampus with the co-activation pattern with the whole brain across studies which were archived in BrainMap. So it's actually not the co-activation pattern of individual subjects, but across study. The second modality was resting state functional connectivity. Here we measured the co-activation pattern of the hippocampus with the whole brain across time in subjects. The third one was structural covariance, which describes the covariation of hippocampal gray matter intensities with the whole brain gray matter intensities across subjects. And here, this one is a group-based measure. We used the data of young adults of the Human Connectome project. And I have also to mention that MECOM and resting state functional connectivity represent both more functional function, and structural covariance, we assume that probably represents more likely co-plasticity. So I used as a method connectivity-based parcellation. So for each hippocampal voxel, I computed the connectivity to every other brain voxel in the gray matter, just using the Pearson correlation. So I end up having a seed by target matrix containing for each hippocampal voxel a specific connectivity profile. But here you can see we still can't say whether voxel 1 is really different based on its connectivity profile compared to voxel 2. So what we do is we apply a clustering algorithm, and in my case it was K-means, dividing the voxels based on their dissimilarity and in connectivity profiles. So we end up dividing the hippocampus into subregions, and we assume that the voxels within one subregion are more similar to each other based on its connectivity profile, but dissimilar to the subregion or cluster of the other one. And you can also see here that that each of the subregions then has also a unique connectivity profile to the whole brain. So my first study showed that we can, based on functional connectivity, divide the hippocampus into the anterior-posterior dimension. If we go up into high granularities, then we actually can also see a differentiation pattern in the medial lateral dimension. And these both functional measures are similar to each other. However, structural covariance deviated from it. We still see a head hippocampus anterior part, but if it goes into the body and posterior hippocampus, we actually have a differentiation in the medial lateral dimension, which reminded us of the cytoarchitectonic differentiation into CA 1 to 4 and subiculum. So what we learned from study 1 is that we can actually, depending on the differences and connectivity measures, differentiate the hippocampus along anterior, posterior, and medial lateral dimension. So in my next study, I was interested whether there are any differences in the lifespan, across the lifespan and in dementia. And here I focused on one modality, it was structural covariance. And I had 5 different groups. So we had young adults, middle-aged elderly, then participants with mild cognitive impairment and dementia. And the data came from 6 different cohorts. So depending on the age group, we assumed that the structural covariance would capture some different mechanisms. So in the case of young adulthood, we assumed that it would probably capture co-plasticity, whereas in elderly and in dementia, it might reflect more co-atrophy patterns. So what we found out is that, first of all, we could replicate in the younger group our structural covariance pattern of the first study. And what we see is that these patterns are actually age dependent. And we had the impression that the differences were especially evident in the posterior hippocampus of the green lateral subregion, moving from the tail, approaching into the direction of the into the anterior direction. But this again, it reminded us of this subfield model. However, in dementia, yes, we had again decrease from the tail, but on top of this, we had an increase, or the cluster moved into the medial direction, which we had the impression covering almost the whole body. And this again reminded us of the functional differentiation. And when we compared these different cluster solutions, then we actually showed that this functional model was more similar to the dementia hippocampus. We went one step further and we wanted to know what are actually underlying structural covariance pattern. And so we did it for every age group. What was striking is that in the elderly, we suddenly had an expansion of the blue medial subregion. In the dementia group, we had a decrease of structural covariance for the anterior hippocampus. And compared to the elderly, we saw that the green structural covariance network changed, expanded, especially in the frontal and temporal regions. So in order to understand this, if there is any relationship to function, behavior, we did a behavioral characterization using Neurosynth to understand, can we actually describe these networks? So what we saw here is that in the young group, we have the anterior hippocampus associated with a lot of behavior related to perception, emotion, cognition, and something like regularity, reactivity, which indicates maybe an automatic response. What was interesting is that in elderly, we suddenly have this blue association with motor related behavior, but also with language, episodic memory, etc. In dementia, however, we had the impression that the green cluster and also its associated network was suddenly related to higher functions such as comprehension, language, photography, theory of mind. So to sum up very quickly, we had the impression that aging is reflected in the hippocampus, especially in the posterior part. And because of the poor behavioral characterization of this green hippocampal lateral subregion, and based on its actual structural covariance networks, we speculated whether it has something to do with white matter or something related to the covariation of white matter of the cingulum, the fornix, and actually a recent study also showed that in aging, these white matter tracts related to tapetum, dorsal hippocampal commissure, posterior cingula are actually affected and decreased. And on the other hand, we had the impression that this blue cluster was more related to the inferior longitudinal fasciculus based on its behavioral characterization. In dementia, however, we saw that the hippocampus follows more a functional division. And based on its structural covariance network and its association with behavior such as language, comprehension, orthography, we speculated whether it has something to do that some pathogens, amyloid plaque or neurofibrillary tangles, are actually spreading across the functional networks. So to sum up here, we learned that the hippocampus structural covariance pattern changes or differs across the lifespan and in dementia. We hypothesized that aging is more affected in the posterior hippocampus, and in dementia, the hippocampus follows an anterior-posterior differentiation patterns. So then I went into childhood and adolescence. And again, I focus on structural covariance because I think it's a very exciting modality. And we had 3 different age groups. So late childhood, between 6 and 10 years, early adolescence between 11 and 14 years, and middle adolescence between 15 and 18 years. We had 3 different cohorts, CMI, HBM, NKI, PNC. Here, interestingly, we found that we have, again, in late childhood, an anterior-posterior differentiation pattern, which we know from dementia. But in adolescence, we see the previous pattern, which we also saw already in adulthood. If we look into the underlying structural covariance networks, then we see that in late childhood, the anterior network is expanding. What we see for the green cluster is that only in childhood it seems to be expanded, but especially in the posterior brain. And if we go to the blue network, then we see that it's actually expanding with higher age. We again characterize them using neuroscience behaviorally because we wanted to know, is there any association with behavior? And we saw that the anterior hippocampus is basically related to a lot of behavior, which is not so surprisingly. But here in adolescence, in early adolescence, suddenly the blue one is related to motor behavior and motivation, which is actually not evident, the motivation association in the middle adolescence. We went a step further and actually wanted to know, okay, is there any genetical profiles that are very specific to these networks and subregions? At this point, I want just to say, yes, they are. And to summarize them very quickly, for the anterior hippocampal structural covariance network, we had the impression it has something to do with an automatic physiological response related to blood pressure, for example, pain perception, hormones. Then interestingly, this green network, which was associated in late childhood with the posterior brain, seems to be involved in endocrine stress reaction, memory and learning, in terms of that the genes that were associated were related to synapse formation, guidance, and neurogenesis. And our last network was very nicely reflected in the gene profiles, being related to action-oriented and motivational network, such as attention, dopaminergic pathway, reward, pain, incentive. So one last slide here. Because I had the impression that something is going on in this posterior hippocampus in aging, but also here in late childhood, I was interested whether we would also see it in gray matter volumes. So what we did is just we looked into the anterior and posterior hippocampus gray matter volumes, and we saw that for the posterior, but not for the anterior hippocampus, we have an increase of gray matter volume between the age of 9 to 11, which is very nicely aligned with previous studies showing that there is a second increase of neural growth just before puberty. The anterior hippocampus is very important in late childhood, which is also in line with the recent study showing that it is associated with memory and language. And the posterior hippocampus, especially the body part, seems to be more related or comes to the forefront in adolescence and seems to be related to motor reward system, which is also very nicely aligned with previous studies. What we learn here is that in late childhood, The anterior hippocampus seems to play a major role, whereas in adolescence, the more posterior hippocampus, especially the medial part, seems to pop up. So I want to thank Sarah Genot, Simon Eickhoff, and my current group. Thank you very much for your attention. And if you like the talk and would like to recommend me, you can do this on Women in Neuroscience Reprogram. Thank you very much. And
thank you, Anna. Just maybe FYI, we asked speakers to quickly paraphrase questions coming from the in-person audience, just because we still have some audio trouble with the questions coming from the audience. So that you're not surprised. So yeah, that being said, any questions from the in-person audience?
Thanks, Anna. I was wondering about the sex differences in the development group.
Yeah, we did not look into it. So we did it years back for functional connectivity and a little bit of structural covariance based on the human connectome data, and we could not find any sex differences. So we stopped at that point. But I think for especially adolescents phase, it might be an issue. So I look into just the gray matter volume. I don't see any differences, but it does not mean that there won't be any based on structural covariance. However, I'm also a little bit, because structural covariance is based on a group, it's a group measure. So if I divide my sample into smaller sample sizes, I'm a little bit unsure whether I will catch up more noise potentially. So I'm a little bit cautious with dividing the groups and going into it, but it's definitely a big issue. And I think also, especially in terms of puberty, and what we also saw is that we have some gene profiles related to the different structural covariance networks that seem to be associated with hormones. So it's definitely an issue to look into it. Yes.
I was curious if you could speak a little bit more about the differences that you find in the parcellations that you get from the functional versus the structural covariance methods. And specifically, do you think that's to do with the functional resolution being coarser than the structural resolution, or do you think it's something about what it's measuring?
That's a good question. So the resolution for the functional resting state data, at least, is 2 millimeters, whereas it was 1 millimeter for the gray matter, so for structural data, of course, even though we resampled. But I would like to say that it has something like a technical issue, but what makes me think of is that we find a very similar pattern of anterior-posterior division, not only on resting state data, but also based on MECAM, which is a meta-analytical co-activation measure across studies, and they have, of course, different resolutions, right? So based on this, I would tend to say that there is something related to function. I hope that we are capturing something functional in resting state and MECAM. But on the other hand, in terms of gray matter intensities and how much it is related to potential cell body decomposition in voxels, that might be definitely a reason why we find this differentiation. Definitely, probably.
We also have some questions from the online audience. First of all, thanks for an interesting talk. Is the functional change in the hippocampus that you see in demented subjects versus normal controls associated with volume loss in your sample? I.e., is the green area shrinking in your demented subjects?
Yeah, I see. So it's not only shrinking, it's like decreasing from the tail, and the blue cluster is increasing, like going. So it's like, it's not just that we have a real like loss of volume. We checked for this. So I excluded subjects where I had the impression if they had 5% more of the voxels that did not have the data in it, then we excluded them. Because we have this kind of shift in the posterior part, I would assume that it is not related to pure volume loss or atrophy, but it's really the core atrophy pattern, because also for the blue brain region, for the blue hippocampal cluster, we see a very clear behavioral pattern. So for this, I assume it has something to do with the longitudinal, like white matter and longitudinal, inferior longitudinal fasciculus, which we do not see for the green cluster, which might more related to this volume.
Okay. One last question. The structural clusters obtained in dementia at early ages seem similar to the clusters you found in the functional clusters of your first study. So for example, following a posterior anterior distribution. Why do you think there is that similarity between structure and function in early age and dementia, and a difference in the other cases?
That's a very good question. I don't have a really good answer for that. I was also wondering, like, how could this actually happen? I was very surprised when I looked into late childhood and saw this, functional anterior posterior differentiation. So I think it maybe has something to do what we also see with gray matter volume across the lifespan, that we have an inverted U shape, right? So we have, like, it seems to be that something that we see in later aging appears already in childhood. So I was wondering whether it just shows that something is happening, that something has to be established, maybe also something related to white matter tracts. And this is one possibility, but there was also another study which looked into structural covariance and language ability, and they found that language, the functional connectivity was prior established before the structural covariance. So it might, so now the question is, okay, what was first there? Was there a functional training established in structure, or was the structure first there on which on top function is built up? So I think we can't answer this question in detail, but maybe some ideas for future where it could go. Thank you.
Thanks.
So next up is Dr. Tammy Tran. She will, she's a postdoc at Stanford University, and she will give a talk on molecular biomarkers underlying neurodegeneration in aging.
That works, yes. It's not good. Down. Okay. Hi. Okay. Thank you so much for that lovely introduction. And I just want to thank everyone who's been organizing this conference. It's been going on so smoothly, and the results have been really interesting. And it's so nice to be back in the MTL universe, I think. I don't know why anyone studies anything else. Okay. So today I'm going to be talking about biomarkers underlying neurodegeneration and memory and aging. And I think the biggest question when we're studying memory and aging is, what are the factors that contribute to individual differences in memory function as we age? And there's a whole host of variables, you know, genetics, brain function, and of course, lifestyle variables such as sleep and physical activity. And what we're really interested in as a group, I think, is how individual differences in brain structure, both at baseline and over time, might contribute to memory function. And of course, preclinical biomarkers such as CSF p-tau play a clear role in memory function. And so today, I'm going to be presenting some data from the Stanford Aging and Memory Study. And this is a cohort of 200 cognitively healthy older adults that have been deeply phenotyped. So they do, functional neuroimaging, they do 7T MRI, and we have blood work on them. And the goal is to try to understand what are the predictors of memory function. And so, and today I'm going to be showing you some data looking at CSF and individual differences in brain volume. And so we all know, and so just as a recap, even though I think we all know this information from the postmortem work, what we believe is that tau neurofibrillary tangles start in the lateral spinal cortex, and that these neurofibrillary tangles then spread to the medial entorhinal cortex before entering the hippocampus. So tau actually moves into two key CA1 sublayer differences. One is the CA1 stratum radiatum, the stratum lacunosum, and the stratum moleculare. And this is often seen as a dark band in the hippocampus. And so I'm just going to refer this during my talk as the SRLM, because unfortunately, even in 7T MRI, we're not able to dissect these sublayer differences. And then following this, tau neurofibrillary tangles then moves into the somata of CA1 neurons. And this is going to be located in the stratum pyramidal cell layer, or SP. And following this, tau moves into other connected hippocampal areas, such as the subiculum and other connected cortical regions. And so this leads me to my two research questions. One is, how do early structural changes in the medial temporal lobe relate to Alzheimer's disease biomarkers, such as CSF p-tau, in cognitively healthy older adults? And the second question is, how do these biomarkers and individual differences in volume predict memory performance in this preclinical population? And so just to talk about the participants a little bit, we have in our baseline visits 200 cognitively normal older adults that did high resolution 3T and 3T fMRI and MRI. We also did 7T on all of these participants, CSF, genetics, and neuropsych. In 2020, we added tau PET on a quarter of our participants. Unfortunately, the pandemic happened, so we weren't able to collect the whole set. In 2022, we are now doing our 7.5 to 8 year follow-up, and we're just tracking all of these participants over time and repeating all of these baseline visits as well as tau PET on these participants. And so today, I'm only going to be showing you data from the baseline visit. And so for participants who did 7T and CSF, there were 147 older adults. We recruited participants between 60 and 88 years of age. Our average is about 70 years of age, and you can see that this is a pretty highly educated and white population. We are in Palo Alto, San Francisco, and the surrounding Bay Area, so not too much diversity in the type of older adults that want to come in for a 3-day visit, unfortunately. But then if we move on to our 3T, T2 segmentations, what we did was segmented the head, body, and tail for all of our participants. And in the body, we then segmented the hippocampus into the different subregions, DG, CA3, CA1, and subiculum. And in terms for lateral and medial entorhinal cortex, we took inspiration from the original Brock and Brock work. I don't know if you can see because it's a little bit blurry, but here. But you can see here that this is entorhinal cortex, which we then translated into medial entorhinal cortex. And then for the transentorhinal cortex, we then translated that into lateral entorhinal cortex. And so given the space that we are in right now, I just wanted to take a moment and talk about the fact that, as we all know with the hippocampal segmentations, for the entorhinal cortex, as we're moving further into there, we're all trying to delineate the entorhinal cortex, but there's a large amount of variability across these different segmentation protocols. Sometimes we're using different terminology. There's a great deal of overlap, but in terms of volumetric analyses, in terms of functional analyses, these are going to clearly impact our results and our interpretations, and especially moving forward in the data that I'm going to show you today. Okay, so if I look at lateral entorhinal cortex volume and age in our participants, what you can see is that lateral entorhinal cortex volume is correlated with age on the left hemisphere, but is not correlated with age in the right lateral entorhinal cortex. If we add p-tau into this relationship, and all of our models do control for age and sex. And then when we're looking at memory, we include education, and all of the volumes are adjusted for intracranial volume as well. And so you can see that when you include p-tau into this model, lateral entorhinal cortex is not significantly correlated with p-tau in this population. And so if we actually look at the spread of our data here, you can see that the lateral entorhinal cortex volume spreads pretty widely between 350 millimeters to 800 millimeters cubed. And so you can see when I was going back and I was looking at this variability in the lateral and the medial cortex anatomy, you can see that because of the depth of the collateral sulcus, there's just going to be a large amount of variability across all our participants. And if you look in the literature, I think a lot of this data that has found significant results has looked at thickness, which you can kind of ignore the anatomical variability, or you can look at baseline difference measures in which you're accounting for this anatomical variability. And so that's what we're hoping to do next in our follow-up. Okay, but now I'm going to move on to CA1 volume, which is the next region affected by p-tau. And we can see here that left CA1 volume and right CA1 volume are positively correlated with age, decreased volume as we get older. And in terms of p-tau, what you can see is that when we're controlling for age, p-tau is still significantly related to volume in the left CA1, and that this is not statistically significant in the right CA1, but there is sort of this trend in this direction as well. And so the first question of I've answered the first question about how early structural changes in the medial temporal lobe relate to p-tau. And so the next question is trying to understand how p-tau and volume in CA1 relate to memory performance. And so we looked at memory performance on two separate tasks. And the first task is an associative memory task. This was actually done in the fMRI scanner, and we previously published some of this data. But in this study, participants were asked to learn a series of word picture pairs. So learn violin and a famous face like Queen Elizabeth, or a word, bike, and then a famous location. A test phase, what we did was that we showed them the word cue, and they had to try to tell us and remember the picture that was associated with it. And so, so Ali Chell has kind of published some of this data looking at MVPA, but I'm not going to touch on it today. And so one big question that we had was, how does p-tau relate to performance on this task? And what you can see is that increasing CSF p-tau performance measured as associative D prime decreases. So as expected, older adults do perform more poorly when they have increased levels of p-tau. And so, just for visualization purposes, we've also split up the participants into amyloid negative and amyloid positive. And you can see it's the amyloid positive, tau positive participants that are really drawing this correlation. Okay. And so then next we looked at the relationship between CA1 volume and performance on this task. And you can see that across the board, as CA1 volume increases, performance on this task also increases. And if we add p-tau and CA1 volume into this relationship, you can see that p-tau actually accounts for all of the variability that is being associated with CA1 volume. So in this case, CA1 volume does not seem to contribute unique variance in predicting this relationship with memory. So the next task that we did, it was a mnemonic similarity task. And so this was done outside of the scanner. And if you're not familiar with this task, I'll breeze right through it. But the encoding phase is you see a series of objects, and you have to identify these objects as indoor or outdoor. And later on in the test phase, you see the three options are old, similar, and new. So you might see new items, which you clarify as new. You might see items that are old, so old items that you previously saw. And most critically, you kind of see these, you see these trials in which there is a slightly similar object to what was previously presented. So the shoe is slightly different from this previous shoe that was shown before. And you also see that this pineapple is slightly different if you look very carefully than the pineapple that was previously presented before. So your response there would be similar. And so embedded in this task are several key difficulty trials. And so the trials, the stimuli range, and so we call these Lurbins. They can range from the most difficult and the most similar. So pineapple 1 and pineapple 2 are very similar to more easy trials in which the items are less similar. And so as expected, if we look at performance across all of these different similarity bins, you would see that performance improves as the task gets easier, right? And so participants at level 1, the task is very hard, so their D prime is relatively low, as opposed to at the least similar items, level 5, where the performance is much higher. And so one big question is, how does p-tau relate to this? And so we had previously looked at this. And so this is kind of a complicated graph, so I'll walk you through it really quickly, but this is a linear mixed model, which indicates that every single participant is represented 5 times. So for each of the LIR bin, so 1, 2, 3, 4, and 5. And you can see here that when participants are at low levels of p-tau, there is this improvement across all of the LIR bins as you would expect. So as the trials get easier, their performance gets better. In comparison, if we look at the participants with high levels of p-tau, you do not see this improvement across the performance. So participants with LIR bins 4 and 5, they're performing just as well as they did on LIR bins 1 and 2. And so this indicates across the board that participants with p-tau are showing an interaction with similarity trials. So they're performing just as well as they did with the older adults with low p-tau, but it's not until the trials get more easy where you would expect improvements in performance that they're not really showing that gain. And this is actually relatively similar to how MCI participants perform on this task. So this is some data from Craig Stark's group, and you can see here, so the data is represented a little bit differently, but we do have the LIR bins down here. And this on the y-axis is going to be 1 minus the probability of old. So just think of this as a metric for performance. And you can see at LIR bins 4 and LIR bins 5 that MCI participants are once again struggling with this task. So what we're capturing in our cognitively normal older adult sample, that participants with p-tau are performing similarly to MCI participants. And so one big question now is, what about volume? And what we see, if we look at left CA1 volume, is that there is an overall gain across the performance. And so there's a strong main effect that we find in which CA1 volume, independent of p-tau, because we're controlling for p-tau in this model, improves performance on this task. And of course, when you're looking at the MST task, everyone always wants to talk about left or DGCA3. And you can see that in DGCA3, the same pattern of results holds. So these participants with left DGCA3 controlling for p-tau also show gains in performance on this task. So in summary today, what I've shown you is data from 2 memory tasks. One is this associative memory task, in which is kind of a retrieval task. We're really trying to encode and retrieve episodic details. And this is hypothesized to be more directed towards looking at CA1 function because of this mismatch detection that CA1 is hypothesized to perform. In comparison, we also looked at the mnemonic similarity task. And this task, in which DG is hypothesized to perform pattern separation, separate and orthogonalize those different representations, and while CA3 is hypothesized to perform pattern completion. And so in summary today, what we've shown is that in p-tau, it's associated with lower CA1 volume, and it's also related to poor memory performance across the board. But if we look at structure, CA1 volume and DGCA3 volume is associated with poor performance on these 2 memory tasks. And it's kind of this confusing relationship in which it looks like MTL structures, at least in cognitively normal older adults, appear to have an effect on memory performance that is independent of p-tau. But at the same time, we know that p-tau is affecting these volumes in CA1 simultaneously. And so it could be that in our preclinical populations, we're able to kind of disentangle this relationship a little bit more. But if we're looking at MCI, the relationship between volume and p-tau is so much stronger that we're not able to look and see how volume relates to memory performance. And so I think I'm out of time right now, so I'm just gonna the normal part. We also did 7T data on these analyses, and we just wanted to say that we know that CA1 doesn't change uniformly. And so we're looking at CA1 sublayer differences, specifically identifying the stratum pyramidal and the SRLM region in all of these participants. Happy to talk about this data later, but I will move on. Okay, and so I just wanted to summarize that, really what the goal of SAMS is trying to do is try to understand strong individual differences in memory and trying to see how we can predict memory changes in older adults and how could these individual differences in memory early on in aging be affecting the preclinical progression onto Alzheimer's disease. Okay. And I just wanted to thank the SAMS team, all of the people who collected my data, my funding organizations, and tell you we are looking for postdocs. So come join the team. We're all still here, but we want more people. So if anyone's interested, please talk online too.
Thank you so much, Tammy. All right, well, we are unfortunately a little over time, so we will actually prefer the online question that we have because we will still be here after the symposium. Yeah, so. Okay, first question. Hi, Tammy. Exciting study. You found the relationship between CA1 volume, left, and p-tau. Was this specific to CA1 or seen with other hippocampus subfields?
Oh my gosh, now my secret slides can kind of play a role here. Okay, so the next volume that is affected by p-tau is going to be subiculum. And you can see here that subiculum has a correlation with age, but in terms of p-tau, there is no relationship between p-tau and subiculum. If we looked at total hippocampus, this is true. If we looked at left and right hippocampus, we're just not really seeing a relationship with p-tau in any other region except for CA1. Yeah. And we also looked at head, body, tail, anterior and posterior as well.
Okay. All right. Thank you so much. Next up, we have Dr. Thanh P Doan, who is a resident and also affiliated with the Kavli Institute in Trondheim and also the Norwegian University of Science and Technology also in Trondheim. And he will give a talk on functional and comparative neuroanatomy of the parahippocampal region.
Hi, so first, thank you very much for the kind introduction and the organization. It has been really wonderful to come here and to get to know you. I have a little disclaimer. I'm actually a clinician for the past 4 or 5 years, so I'm doing less research. And what I'm going to present today is more a conceptual talk. So there is some data, but everything is published. But it's more, if, I've been talking a lot with Menno, with whom I took a PhD, to see whether the findings we did during my PhD could make sense when it comes to the functional organization of the parahippocampal region, if this makes sense to integrate all these elements. So I have basically 3 parts. It's a very big topic and a very ambitious title, I think. So I'm sorry, I hope I don't offend any of you by repeating very simple things. And I hope I also don't go too fast over very complex elements. Anatomy is quite tricky. But as you know, we have something that has been described as a parallel path in the parahippocampal region. We know that there is basically 2 pathways. It's a little bit discussed, but we know we have something called the WAD pathways that is basically coming through the perirhinal cortex to the lateral entorhinal cortex. And we have this WER pathways providing spatial information from the parahippocampal cortex to the medial entorhinal cortex. Now, PHC is the human primate nomenclature, but in rodents it's not called, but the homologous structure is called the postrhinal cortex, so it would be POR. And I don't want to repeat that too much here because I think you know all of this. What I want to speak about today is a little bit how we can challenge with new elements that have been found lately, this model, and if it can help us maybe to build a little bit more translation, something that I'm quite interested in, being both a scientist and a clinician. So when we want to define an anatomical area, we can look at the localization. We can look also at the architecture, whether it's in an MRI with different sequences or so on. But the histology as well, there is different components. We can look at the cellular architecture, cytoarchitecture, the chemo architecture, the genome architecture. And this gives us additional elements in order to delineate these different areas. But we can look at the connectivity, both the input to the areas, the intrinsic connectivity, and also the extrinsic outputs to other areas, which is particularly relevant in the parahippocampal region. And at least the functional signal, what do we find here? So we will focus first here on this entorhinal parahippocampal region. So if we take a look back at the evolution, basically what's interesting is to see that the hippocampal formation is highly preserved across vertebrates. I mean, from fish to humans, you can just look at the slide. The structure is very comparable. There is basically an expansion of the cortex, and the hippocampal formation slowly moves caudally and then more medially into the temporal lobe when the cortex gets more and more gyri. But this structure is very well preserved. When we look at it, basically, we see the hippocampal formation, which is composed of the dentate gyrus, CA3, CA2, CA1 field, and subiculum, if we take the hippocampal formation. And it has more or less 3 layers. And then there is this area of transition constituted of the pre-parasubiculum entorhinal cortex, which is sometimes called the perialocortex. It's quite debated, but let's call it like a gray area before we get into the isocortex, neocortex, composed of other brain areas such as perirhinal cortex when you are and parahippocampal cortex caudally. So in the hippocampal formation, as you know, there's been discovered place cells. These cells are basically located into the hippocampus. And what is interesting is that they're distributed across a gradient. It has been shown in rodents, in monkeys, and even in humans. But what is interesting for today's talk is that it has been shown recently also in birds in a very interesting paper in Science last year. And it shows basically that spatial signal, this place signal, is highly conserved also into species. And it has the same functional gradient distribution. Now we know that this signal is not generated within the hippocampus. You can basically section the fibers between CA3 and CA1. It has been done already in Trondheim. And the place signal does not disappear. And it was basically the finding that led people to reevaluate that these inputs that are relevant for place signal might come from the neighboring entorhinal cortex. And we know there is a gradient of distribution as well, going from the dorsal part of the entorhinal cortex in rodents to the ventral part, that also follows the same hippocampal gradient. It holds true for humans as well. When colleagues at the Karolinska Institute probed the activity of this region in the entorhinal cortex, they found another very interesting place signal called the grid cells. And what is interesting for today's talk is that the gradient is distributed also functionally between the entorhinal cortex and the hippocampal formation, meaning that in the dorsal part of the entorhinal cortex, that would slightly be homologous to the closest part of the collateral surface into the entorhinal cortex in humans, in primates. We have grid cells basically that fire with very little distance close to each other. If you are not familiar with grid cells, I'm sorry, it's cells that basically are active in a very regular fashion and tessellate the entire environment. And they're found in the medial entorhinal cortex. So basically, these grid cells and place cells, they are highly dependent. If you numb out the medial entorhinal cortex, the place signal will change. If you numb out the place signal, the medial entorhinal cortex grid cells will change. And similarly, this cell, the grid cells, they are highly conserved into evolution. We can go back to this kind of cladogram here, and we can see that it has been found in different species, including bats. So it started in rodents, then monkeys, it has been found, slightly different fashion. In humans, it has been found in epileptic patients with invasive electrodes. But in bats, it has been well as found. And it's interesting because bats, they have a common ancestor that is more primitive than rodents. So likely, this grid signal is something that is very well conserved and that arose at least in a very early mammal. Now in LEC, we don't have this grid signal. we have a very complex signal that we do not really know what it's doing. That's why it's called sometimes a what signal, but it's believed that it codes the content of the experience, the ongoing experience. Sometimes it's described as a temporal signal. Now, there has been a lot of papers, but I picked this one. It was from Howard Eichenbaum group in Journal of Science in 2016, because you can look at these typical cells into the medial entorhinal cortex or lateral entorhinal cortex, but actually both cells, both at the single cell level and population levels, they code both for spatial feature and non-spatial feature. And it's really challenging because basically we can't really say that LEC is providing non-spatial information while MEC is providing spatial information. There is already a high level of integration within the entorhinal cortex. Then I will turn to part 2. What is not really talked about too often, I think, but very interesting finding is that if you look back to older species, such as lizards, sauropsids, basically lizards, fish, you can see that not only they have a homologous structure to the hippocampal formation, but they do have also homologous structure to the entorhinal cortex. And these structures can give us information about where they rise. And when it comes to the medial entorhinal cortex and lateral entorhinal cortex, actually they seem to originate from 2 different, very distinct embryonic parts. So the medial entorhinal cortex seems to originate from the medial pallium, which is related more to the hippocampal formation, while the lateral entorhinal cortex seems to derive rather from the dorsolateral pallium, which is more of a neocortical structure, which is very interesting because they look so similar, but actually they have distinct origin. What has been shown also in 2018, Flavio Donato, in really nice paper in Science, is that the medial entorhinal cortex is actually driving the maturation of the lateral entorhinal cortex. And it's quite interesting to see that maybe something that is more similar to the hippocampal formation kind of basically leads to the maturation of a neighboring more neocortical structure, which end up to be quite similar. So why is it relevant to us? It seems that they have different origins, right? We could use that maybe, to speculate about what we could call MEC and LEC in primates, including humans, of course. But what's used in rodents often, at least when it comes to tracing, is that you can use the hallmark of entorhinal cortex, which project to the hippocampal formation in a very specific fashion. You have the medial lateral path, perforant path, and the lateral perforant path. I won't make use the offense just to repeat these things, but there is this paper from Meno in 1989, which shows basically that they project distinctly to the dentate gyrus in CA3 and also to the subiculum CA1. In rodent, it's quite clear cut, but when you turn to the primate and you go through this evolution, I think it has been shown in humans with fMRI-beta in the group of animals, you can see basically that it looks more and more of a gradient, and it becomes very hard basically to base your assumptions that you have an MEC equivalent in humans based on the same criteria as rodents. So I will move quickly on to the intrinsic connectivity of MEC and LEC. Can you use this? Well, it has been shown in MEC you have a very specific way of organization intrinsically, is that you have these principal cells that are connected through inhibitory interneurons, and they have a very hardwired intrinsic connectivity. Basically, that is possible to probe with electrophysiology, where you can basically decipher intrinsic connectivity and see whether this is coding high-level representations or low-level representations. Recently in Nature, Richard Garner published with colleagues this beautiful paper where he described that all grid cells in medial entorhinal cortex sit on the torus, which is a topological form, which is quite well defined. Now we believe that it was because the intrinsic connectivity of the medial entorhinal cortex is so peculiar, but in LEC you do have the same, although you don't have grid cells. So that's a big thing that is quite strange to us, because MEC and LEC actually, when you look at the way they're connected intrinsically, they are very similar. So why should one have grid cells and not the other? When you look closer to the electrophysiological properties of single cells into the medial and lateral entorhinal cortex, you don't see that you have any borders, functional borders, which looks like the entorhinal cortex is actually a functional continuum. It's not two distinct entities, even though we would like to call them like this. So now I will just switch very quickly to the last part, the entorhinal cortex. So it's a functional continuum. That's what it seems. I mean, we can delineate, we can discuss about this later. But the real question was, does this difference come from the inputs? Now I will go quite quickly. This is published as well, but basically what we did is tracing injections into rodents, and we look at anterograde tracing from the postrinal cortex, which is the equivalent of the parahippocampal cortex. And what we found is that the postrinal cortex sends massive projections to the lateral entorhinal cortex, which was quite the opposite of the model. We did control experiment with retrograde tracing just to make sure that we were not doing anything wrong. And then we tested whether this new projection we described was actually synaptically active by doing electrophysiological experiments. I'm running a little bit of time, I'm sorry. But basically, we did both optogenetic stimulation paired with electric stimulation in patching single cells into the lateral entorhinal cortex, and found that single cells in the lateral entorhinal cortex in layer 2, actually, where Alzheimer's disease might start, by the way, receive both inputs from the postrinal and the perirhinal cortex. And what was even more interesting is that looking back at all the data in the rodent, but also kind of looking at its flat maps in primates a little bit more carefully, the organization seemed very similar. So it seems actually that in LEC, you have a convergence of inputs. And this parahippocampal cortex, postrinal in rodents, actually projects preferentially to the lateral entorhinal cortex, not to the medial entorhinal cortex. So the question is, what actually does project into the medial entorhinal cortex? Well, it remains a couple of structures called the pre- and parasubiculum, the vitrosplenial cortex. And what is interesting is that they receive basically massive inputs from the anterior dorsal thalamus, which conveys the head directional signal, which is critical for formation of grid cells. And also they seem possibly to provide some speed signal. So these two inputs, the head direction and the speed signal, are very important for path integration and the rise in the brainstem, very primitive structure. I think it goes in line with the fact that MEC might be more primitive. So that's my last slide. And then I'm happy to take questions. That was a little bit conceptual with a lot of anatomical jargon maybe, but if you want to remember something, here it comes. The hippocampal formation and parahippocampal system demonstrates a highly preserved architecture and connectivity across species. I hope we agree. The entorhinal cortex functional signal has been previously assumed to relay spatial, MEC, and non-spatial LEC information, but however, both information, both spatial and non-spatial, are present across the entire entorhinal cortex. So for me, at least, this dichotomy does not make sense in terms of function. I'm really happy to discuss about this. The third point is that the entorhinal cortex seems to arise from two different embryonic structures, which is quite interesting because the MEC seems closer to the medial pallium, which is more closely related to the hippocampal formation, while LEC seems to arise to a more neocortical-like part. Then we saw very quickly that MEC drives the maturation of LEC, leading to similar intrinsic organization, and that within the entorhinal cortex, we don't have clear-cut functional borders. And nearly last, the extrinsic input to the entorhinal cortex is actually what defines its functional phenotype. Is it spatial, non-spatial, and so on? This is, I think, what is critical is to look at the inputs precisely to the entorhinal cortex across species. And these functional phenotypes are basically complex representations. It could be grid cell, it could be the signal, but it's an entorhinal made language, which is read by the hippocampus further. And I think that we believe that's the role of the entorhinal cortex. So the entorhinal cortex is not part of the hippocampal formation. So I hope you will agree with me that the entorhinal cortex is the main integrator of the parahippocampal region, which is not conflicting that it might be the subiculum in the hippocampal formation. But basically, the role of the entorhinal cortex seems to be this interface between the hippocampal formation and the rest of the neocortex. And it seems maybe that there is a small part that we call the medial entorhinal cortex in species that is used as a template to catch a little bit of a neocortical-like part and model it such that it looks similar and such that the entorhinal cortex as a whole can integrate and communicate with the rest of the neocortex. On this slide, I would like just to thank my supervisor, Menno, and Maria José Donatella Gardos in Oslo, Erik Stamland Nielsen in Trondheim, and Shinya Ohara in Tohoku. They helped me a lot for my paper in this experimental paper I mentioned. And the last point is that although I've been working as a clinician for 4 or 5 years, I've been also working a lot to build translation in Norway. I'm trying very hard. And I've been teaming up with 2 really great people, young and promising, Tobias Navarro-Schroeder, where we are working on interesting questions using the 7 Tesla MRIs that we have now for the last few years. And also with Jürgen Sugar, with whom we worked very hard to get access to intracranial recordings. And we are setting up also microelectrodes, get single unit recordings in the only hospital in Norway, which is in Oslo. So with that said, we have both imaging methods and electrophysiology in humans and some knowledge in animals. And I think with this combined knowledge and the wonderful platform given by the hippocampal subfield group, there might be something cool happening. Thank you for your attention.
Unfortunately, again, we are short on time.
Yes.
No worries. So just one question that we can do right now. So anyone from, from the in-person audience?
That was a little bit conceptual, so I understand. Yeah. I'm not an animator, of course. Yes, that's you understood as well.
There's some work in animal models suggesting that there's like Wheelan differences between lateral and medial entorhinal cortex. Does this play but the fact that Wheelan cells are kind of everywhere, is there a difference to an accumulation? Because I think that the animal models have suggested that only Wheelan cells are affected in lateral entorhinal cortex.
It seems that that's the area that is primarily affected. You are right. So in humans, it corresponds to the lateral part in close relation to the collateral sulcus, maybe what you refer as transentorhinal cortex or LEC in your talk. In rodents, it corresponds to the part that is very close to the rhinal fissure, and it's also more into this lateral and rostral parts. Yes, you are right, but Wheelan cells can be found both into LEC and MEC. They do present slightly differently when you look at the cytoarchitecture and chemoarchitecture, but they are present there. There might be some weakness there. I think I would suggest to read the work by Asgair Kobro-Flatmoen at the Karolinska Institute. Pretty much all that I know about, yeah. But he has a great review published recently.
Great, thanks again.
Thank you.
So finally, we have a blitz session scheduled. So very short and quick talks. So actually, while I introduce the speakers, please come up to the front so we can run through everyone smoothly. So anyone who's presenting during the blitz, please come in front. So the blitz sessions will be 4 minutes long. We have 5 speakers, so a total of 20 minutes. And then after all 5 talks, we will have 5 minutes for questions. So our blitz speakers are Dr. Jenna Adamns, who is a postdoctoral fellow at Mike Yassel's lab at UC Irvine. Then we have Iris Bosch, who is a research assistant at Gothenburg University. Then we have Negar Mazloum-Farzaghi, who is a PhD student at the Altman Research Institute in Toronto. And this is awkward. Then there's me. Hannah Gomez, I'm a PhD student at the DZNE in Magdeburg. And Anika Wuestefeld, who is a PhD student at Lund University. All right, thanks, Jenna. Hi,
everyone. I'm Hodgetts for the opportunity to present our work today on entorhinal-hippocampal circuit integrity as related to mnemonic discrimination and amyloid-beta pathology in older adults. Down. Okay, so just for some quick background, the last two talks set us up well for this. Mnemonic discrimination is a cognitive process that enables the differentiation of similar memories into distinct representations. The neural computation that supports this, known as pattern separation, is performed in the dentate gyrus and CA3 subfields of the hippocampus and relies on direct input from the entorhinal cortex via the perforant pathway. Mnemonic discrimination is particularly vulnerable for aging, especially for object stimuli, but the mechanisms behind this aren't still fully understood. So in this project, we wanted to determine how the integrity of the entorhinal-hippocampal circuit, measured with high resolution resting state functional imaging, is related to mnemonic discrimination impairment in Alzheimer's pathology in a sample of older adults who are still cognitively normal. So based on our previous labs task fMRI data and some other labs as well, we hypothesized that object mnemonic discrimination would be related to disrupted functional connectivity between the anterolateral entorhinal cortex and DGCA3, while spatial mnemonic discrimination would be associated with disrupted connectivity between the posterior medial entorhinal cortex and DGCA3. But as we just saw in the last talk, it might not be that simple. We further thought that amyloid-beta pathology would be associated with more so widespread differences in connectivity across the circuit. So to test this question, we had a sample of 64 cognitively normal older adults who performed a mnemonic discrimination task outside the scanner. They had both object and spatial versions. So quickly in the object version, they looked for differences in the object stimuli themselves, while in the spatial version, they looked for differences in the location of the item on the screen. Based on performance on this task, we divided older adults into high performing group and a low performing group in each domain. Older adults also received amyloid PET, and we classified them as either being amyloid negative or amyloid positive based on global SUVR uptake. Finally, we performed high resolution resting state functional connectivity, and using a study specific template measured ROI to ROI functional connectivity between entorhinal subregions and hippocampal subfields. And we focused here on connections between regions with no anatomical connectivity. So first we compared functional connectivity across the circuit between older adults who performed well or poorly on this task in the object version. We found that low object mnemonic discrimination group, shown here in dark red, actually had increased functional connectivity between ALEC and DGCA3, with no other significant differences across the circuit, which supported our hypothesis of a specific object-related mnemonic discrimination deficit here. We next looked at differences between the spatial mnemonic discrimination task groups. And while we didn't find any statistically differences in the circuit, we did find a trend for differences in posterior medial entorhinal cortex to DGCA3 connectivity. Finally, we looked at connectivity differences between the amyloid groups, and we found that amyloid positive older adults, shown here in dark green, also had increased functional connectivity between ALEC and DGCA3, mirroring the pattern we found in the low object mnemonic discrimination group. Further, we found differences within the hippocampus itself too, so lower connectivity between CA1 and subiculum, and a trend for decreased connectivity between DGCA3 and CA1. Finally, we wanted to see whether some of these effects were related to neurodegeneration in the circuit, so we assessed volume using ASHA segmentation. And we found an interesting association and interaction in which the low object mnemonic discrimination group, this increase in connectivity was related to lower entorhinal volume, which indicated the pathological nature of this increase in connectivity. Further, amyloid pathology was also related to lower entorhinal volume, and these were specific to entorhinal cortex, not dentate gyrus or CA3, which suggests this might be resulting from entorhinal dysfunction. Just to quickly sum up, our results suggest that amyloid may indirectly lead to mnemonic discrimination impairment through changes to entorhinal-hippocampal circuit integrity. And this might also help explain the specific vulnerability of object mnemonic discrimination in aging and preclinical Alzheimer's disease. I'd like to thank our lab and our study co-authors here, and I'm happy to take questions after the data discussion. Thank you.
Thanks a lot for having me. In the next few minutes, I would like to present a project that I started working on a couple of weeks ago, where I am exploring volumetric changes in medial temporal lobe subregions in COVID-19 patients. I think it's pretty safe to say that COVID-19 doesn't need much of an introduction anymore. So what is known is that neurological, cognitive, and neuropsychiatric manifestations have been described in a large proportion of patients. This can also be referred to as neuroCOVID, which has a heterogeneous presentation. Although the underlying pathology remains largely unknown, volumetric brain changes have been reported, including in MTL subregions. Previous study has reported decreases in the left parahippocampal gyrus and the left and right entorhinal cortex volumes. And another study has reported higher gray matter volumes in both hippocampi. So these studies, taken together with frequent reports of cognitive impairment, underline the medial temporal lobe as a potentially affected brain region in COVID-19 pathology. One of the aims of our larger multimodal NeuroCOVID study is to explore volumetric changes in MTL subregions. The following three groups are included in our study. First, we have a COVID-19 patient group that was included 1 month after hospitalization. Second, we have a COVID-19 patient group that was recruited due to remaining CNS symptoms, and we have a control group. For group 1, we have baseline and 3 and 6 months follow-up data available. And for group 2 in the control group, only baseline data at the moment, and around 19 to 20 subjects were included in each. In order to obtain the volumetric measures, we used ASHES, and I'd like to thank David Doan for processing our scans. High resolution T1 weighted and MTL focused T2 weighted scans were used as input. And we studied the following regions, looking at each hemisphere separately, and our analyses in general are quite exploratory. I would now like to show you some first results. I'll focus on comparisons between controls and group 1, as we currently only have longitudinal data available for group 1. In order to compare the different regions between the groups at baseline as well as during follow-up, we used the linear mixed model, but we didn't find any differences for all regions of interest. I do want to show you 2 example plots. So here you see example plots for group 1, showing right entorhinal and left parahippocampal cortex volumes over time, where volumes have been Z-score transformed relative to controls. So the mean volume of controls would be 0. And this allows us to look at the p-value of the intercept. But as these plots also demonstrate, didn't find any differences. So these results would suggest that there are no changes in MTL subregion volumes for COVID-19 patients that have been recruited 1 month after hospitalization. I think patient group 2 might be of particular interest. So the patient group that was recruited due to remaining CNS symptoms. Preliminary cross-sectional analyses are indicative of greater volumes of the right subiculum, but we're waiting for additional data before further interpretation. I think in general, studies investigating volumetric changes in MTL subregions are quite limited. Study designs are also diverse and results are inconclusive, but I think a strength of our study is that we use some dedicated method for the MTL subregion segmentations. For future analyses, we'll include studying associations with neuropsychological and clinical measures, and we're very much looking forward to the follow-up data, which will hopefully provide more insights. Thanks a lot. And in particular, I would like to thank my research group and David Biron and Hanna Baumeister for their help.
Okay, good afternoon, everyone. My name is Negar, and the title of my talk today is The Association Between Medial Temporal Lobe Subregion Volumes and Cognitive Assessment Scores in Healthy Older Adults. So previous research has found that volumetric measures of specific subregions of the medial temporal lobe, or MTL, are associated with tau pathology and neurodegeneration related to early stages of Alzheimer's disease. However, it's less clear whether volumetric measures of MTL subregions can identify the earliest signs of neurodegeneration prior to clinical diagnosis of mild cognitive impairment, or MCI, which is a condition that often progresses to Alzheimer's disease. And current manual segmentation methods that are commonly used to measure the volume of MTL subregions are labor-intensive and time-consuming, and so we may need a more efficient yet just as accurate alternative to manual segmentation methods. So this leads us to our current study where we used a powerful automated segmentation software tool called the Automated Segmentation of Hippocampal Subfields, or ASHES for short, to ask the question of whether ASHES can detect the earliest signs of atrophy in MTL subregions of healthy older adults, as inferred by correlations with scores on the Montreal Cognitive Assessment, or MoCA, which is a dementia screening test. Now, our research was motivated by prior work in our lab in which, using manual segmentation and a protocol developed in our lab called the OAP protocol, Olsen, Young, and colleagues found that volumetric measures of the anterolateral entorhinal cortex, which is an MTL subregion where tau pathology consistently accumulates in early Alzheimer's disease, were related to scores on the MoCA in healthy older adults. And we can see that relationship on the screen. And so to assess whether automated methods could produce similar results, we trained ASHES on the OAP protocol and applied automated segmentation to the same group of healthy older adults as in our previous work. So the automated segmentations resulted in 3 subfields of the hippocampus and 4 MTL regions, which included the 2 subregions of the entorhinal cortex as originally defined by Moss, Barron, and colleagues in 2015. So here, just like the figure I showed on the previous slide, I have the relationship between MoCA and volume for each MTL subregion of interest. But to zoom in on the regions of interest, we found a significant positive association between automated volume in the gyrus CA2-3 region and MoCA, whereas this relationship was marginal in our previous work using manual segmentation. We also found a significant positive association between automated volume in the perirhinal cortex and MoCA, whereas this relationship was non-significant in our previous work using manual segmentation. And finally, unlike the findings from Olsen and Young, we did not find a significant association between automated volume in the anterolateral entorhinal cortex and MoCA. And so overall, our study indicated that ASHES was able to align well with current manual segmentation methods. However, the discrepancies between the automated and manual methods for the perirhinal cortex and the anterolateral entorhinal cortex could indicate that some refinement is needed for the delineation of these regions. Fortunately, the hippocampal subfields group is currently working on this, and we will be learning more about these regions in the working group tomorrow. Thank you for listening. All right. Yeah, so hi, you've seen me by now. I'm Hannah. And I'm so excited to present first preliminary results from our project on hippocampal thickness in early Alzheimer's disease. So Alzheimer's disease is characterized by pathological accumulations of amyloid-beta and tau peptides. And in the early stages of the disease, we distinguish preclinical and prodromal AD. They are both characterized by amyloid positivity. However, in the preclinical group, patients are cognitively normal, while in prodromal AD, patients can be diagnosed with mild cognitive impairment, or MCI. Particularly tau pathology is actually associated with local neurodegeneration and subsequent cognitive decline, with the hippocampus actually being among the earliest regions to be affected by tau pathology. However, in most cross-sectional in vivo MRI studies using subregional or whole hippocampal volumetric measures, no effects of neurodegeneration can be found earlier than that prodromal stage of AD. This leaves us with the question if the measures that we're currently using are maybe not sensitive enough to those earliest changes. So to answer this question, we teamed up with Martin Reuter's lab at the DZNE in Bonn, particularly with Kirsten Diers, who's a postdoc there, and he is developing this great pipeline that takes a T2 ASHES segmentation, the hippocampal body to be specific, and provides us with a 3D coordinate system, leaving out the dentigyrus, unfortunately. And this allows us to estimate thickness at 861 vertices.
So we applied this to a subsample of the DELCODE study, totaling 207 participants, including healthy controls, 2 preclinical AD groups, one being completely cognitively unimpaired, the other one having subjective cognitive decline, but being cognitively normal on our established neuropsychological measures. And then we also have a prodromal AD group. So the first question that we wanted to answer was, how are AD pathology and hippocampal thickness related? So we went in with a masked univariate analysis, relating levels of CSF total tau to pointwise hippocampal thickness. And here in red to yellow, you see the regions that are significantly negatively related to CSF T-tau levels. So right there at the distal CR1 proximal subiculum, where we would expect it from the ex vivo data that we know. Next question we wanted to answer was, does median thickness in this specific region differ between clinical groups and healthy controls? So I'll walk you through this slide. So we calculated median thickness in this specific row. We also calculated subiculum, CA1, and whole hippocampal volume. And then we just did very basic group comparisons. So here at the top, you see that the mild cognitive impairment group has quite a bit of neurodegeneration, just as expected. However, the only effect of neurodegeneration that we see is here in that SCD group, where only our new structural measure picks up a significant negative effect. And yes, I do see this effect. I'm very happy to talk about that later. So we do actually see significantly larger CA1 volumes in the CU amyloid positive group compared to negative controls. So with that, the take-home message, CSF T-tau levels are related to hippocampal thickness in the proximal subiculum distal CA1 region. And amyloid positive SCD individuals show lower median thickness in this region compared to healthy controls. And with that, I want to thank everyone involved, especially David Baron, who is my PhD supervisor, and also, as I already mentioned, Kristin Nordin, who is developing this great pipeline. Thank you.
Okay, my turn. I'm going to talk about age-related tau patterns, downstream effects in the medial temporal lobe, of course, and cognitively normal older adults. This is some data that we're working on currently. One of the aims was that we wanted to investigate in this cognitively normal older adults and a subgroup of individuals with low A-beta levels. When it comes to age-related tau PET in the MTL, and whether tau PET deposition drives age-related atrophy in the MTL and also changes in cognition. We used 536 individuals that were cognitively unimpaired from the Swedish Biofinder 2 study, and then 413 individuals were in the low amyloid subgroup. We also used ASHES to get our measures of the medial temporal lobe fields, as shown also there in the top corner, and then PET imaging to quantify tau and amyloid PET in the brain. So first of all, we looked at the association between age and tau PET in the earliest region of tau. So there's entorhinal cortex BA35, as you can see in the figure below, and the association with age. This also shows again the strength of the association. And we see that binding increases in both the whole group and also this low amyloid subgroup with age. Then next, we also looked at the association between age and MTL thickness and volume. And as expected, we see widespread associations between age and MTL subfields, with also similar results actually in the low amyloid subgroup, which was also expected. But then we continue to look at the association between the MTL subfields and actual tau PET signal in this earliest tau region, ERC BA35. And we do see that this early tau PET is associated with subiculum, dentate gyrus volume, and BA35 thickness. And additionally, we see that these associations are amyloid independent. So when we adjust for amyloid, these
associations remain. And we also find in the low amyloid subgroup, so the individuals that have no or very little amyloid, that there's this association between BA35 thickness and tau in ERT BA35. So to put this all together, we then ran mediation models and see that in these three regions, so subiculum, and BA35, that there is MTL tau PET that partially mediates the age BA35 thickness association. This is just an example for BA35, but we do see similar results for subiculum and dentate gyrus. And this is, as shown by this kind of gray area, independent of amyloid that is either regional or also global amyloid. And for BA35, again, we see similar results in the low amyloid subgroup. Then we also looked exploratory at some cognition part, and we see that ERC BA35 tau PET and age, but not the subfield volumes, are associated with global cognition. We also do see, again, a similar mediation between age and now global cognition with our MPEG. That is, again, amyloid independent, but we do not see these associations in the low amyloid group. To summarize, we see amyloid independent associations of age with tau PET, which could indicate that primary age-related tauopathy can be measured in vivo in cognitively older adults, normal older adults, and the mediating role of regional tau PET in age structure and age cognition associations is supportive of downstream effects of PART in this group. This may also support anti-tau interventions in age-related neurodegeneration. I would also like to thank the Biofriendly group and Lara, especially. Picture. Thank you.
All right.
Should I quickly handle that?
Yeah, we have one question actually from Anna to me.
Okay, so Anna asks, very great data, Anna. You focus on CSF t-tau. I guess it is highly correlated with p-tau. Are effects similar for p-tau, to be more specific to AD tangle pathology maybe?
Yes. So yeah, great question. It's also quite obvious to actually go in with p-tau in that group. So yeah, the results look very similar. And also, so I just happen to show p-tau here. We do see a little more of a posterior effect with p-tau in the posterior subiculum. So we have all sorts of fluid biomarkers, and we will explore all of them in relationship to hippocampal thickness. However, actually, something that would also be good to mention is that you're working with this in the Biomarkers study, actually looking at relationships with tau PET. And their results also look quite similar. So that's quite promising, I think.
Thanks. So yeah, I mean, the floor is open. So also questions from the audience to all the, is it 5? Data speakers. Laura?
Yeah, I have a question for Negar. I was curious, you looked at automated volumes. Did you do any QC? Like, did you just get the output and run the analysis, or did you also look at them and maybe exclude or edit? What did you make it?
Because of the mic.
Oh, I can see myself. That's a good question, Laura. At the moment, since we only had 40 older adult participants in our samples, and we were trying to compare to our previous work that also was on the same 40 older adults, we didn't exclude any segmentations at the moment, but just visually inspecting, they were quite good segmentations.
Maybe can I add one quick question to that? So the only, if I remember correctly, so the only region that was more accurate in manual than, or one that was more accurate in manual than automated was the anterolateral entorhinal cortex, right?
Right.
Could that be because there's no clear boundary that we actually can see?
Yeah, that's a really great point. So as many of us know, the anterior and posterior boundary of the entorhinal cortex is a point of discrepancy in the literature. And for example, in Laura's work, the anterior and posterior boundaries between manual and automated methods, especially for ashes, was also a point of discrepancy. So it could be that manual methods are slightly more sensitive to detecting subtle changes in gray matter volume than automated methods are at the moment. But we're working on it so we could have a more harmonized approach to delineating that region. Yeah.
Thanks. Any other questions from the audience?
Yes. I was supposed to have like a negative finding, but didn't really see any effect of COVID on medial temporal lobe. You think it's really not there? Is it maybe a power issue?
Yeah, good question. The question is, what are my thoughts on the negative results? I think, so of course, it can be that there are maybe no differences. I think it's also important to stretch that some studies have found some differences for some regions, but there are also a lot of regions where they didn't find any differences. On the other hand, our sample is on the smaller side. And our patient group is also quite heterogeneous. All that while reported percentage changes are quite small. So it might be a power issue. Also, our COVID-19 patients are quite mild in their neuroCOVID symptoms. So that may also play a role. So they didn't show major brain inflammation, for example. Yeah.
Thanks so much. Oh, also question. Yeah, so I'm not sure, maybe I didn't catch it as you were presenting it, but when it comes to cognitive effects in these individuals, is there any data there where you could, for example, look at the association between volume and having had COVID, splitting up people into those who do show any kinds of like long-lasting effects on cognition compared to those who might not show that? So you can kind of dig more into subgroups.
So the question is whether we can explore subgroups in our patient group that show cognitive impairment. Yeah, the answer is yes. We do quite extensive neuropsychological testing, and we're also very interested to maybe look on a more individual basis or creating subgroups. Yeah, so we'll definitely do that.
Any other question? And maybe a short question for you, Jenna. So you showed that, yeah, maybe. You showed that increased functional connectivity actually was related to lower mnemonic discrimination performance, right?
Right. Mm-hmm.
Do you have any thoughts on that, how you interpret that?
Yeah, we're thinking it's probably aberrant network activity because amyloid's been shown to be associated with hippocampal hyperactivity and also entorhinal hyperactivity in other studies. So that's what we're speculating, but we'll have to look into it more.
Great, thanks.
Follow up on that question. So that increased connectivity, could it be driven by predominantly increased hippocampal activity, or did you also see increase of EC activation too?
So we could look at pure activation because it was resting state, so we didn't have anything to contrast it against, but we want to follow that up in other task-based fMRI studies.
Look at the resting state. Well, I tried doing this, but you have a larger sample. You can look at the resting state in EC versus activation during the mnemonic syndrome.
Yeah, unfortunately, they did the task out of the scanner too, so we don't have the data on the same sample right now. So that would be really fascinating though.
It's also directly related to that, so I hope it's going to be quick. You also had something about the CA1 in there, and it was all quite quick, so I couldn't quite see it, but it looked like it was decreased connectivity. So yeah, just be interested in kind of hearing your view on this difference in terms of like increased versus decreased connectivity on the basis of like subfields and
Absolutely, yeah. So the question was, there was a finding in the study that the connectivity between CA1 and subiculum was decreased with amyloid. And that was a little bit surprising to the opposite direction, but we had found a study, I think it was an animal model, that showed that sometimes subiculum can downregulate its activity when DGCA3 is upregulated. So it could be potentially some type of compensatory finding, but definitely need to investigate that further as well.
Sure. Yeah, so then I think if there are no more questions, thanks a lot for the in-person audience here, but also thanks a lot to everybody on Zoom who is still here. We will now close the session here and we'll continue with the reception outside. And maybe that you're clapping, just again, a big thank you to Hannah, Anika, and actually